Okay. Welcome, everyone. We're ready to start. Tech is working. Welcome, as I was saying, and thank you very much for being here. It's a pleasure to see you all today here, and I hope you enjoy our symposium on engineering biology that we are running today. As you have seen the program, we have a very exciting list of events throughout the day, ending with a highlight annual lecture for our institute. So we are IMSE, the Institute for Molecular Science and Engineering. We are one of the Global Challenge Institutes of the college and work with a remit to facilitate and promote interdisciplinary work throughout the college and beyond. And so in IMSE specifically, we are interested in problems where molecules and understanding innovation at molecular level has an impact at engineering and function level. So that's what we do. The institute is very broad in what we do, and each year we highlight different areas that are exciting and are up and coming, and where grand challenges are yet to be addressed and discovered. And it's really a pleasure, as I said, to have today a day of events on engineering biology, which really is a perfect point of highlight of how interdisciplinarity between areas has a lot to contribute to future science. So thank you, all, as I was saying, all of you for being here. A special thanks to all the speakers of today. I hope you enjoyed the day. And before I introduce our first speaker, just to tell you this is a hybrid event. Tech is working. There will be questions after the talks as usual. And for those of you joining us online, please put your comments or your questions on the comments box, and the team here will pick them up. Okay? So with that, I can't remember if I introduced myself or not. Did I introduce myself? I think I did. Anyway, in case I didn't, because sometimes I forget, I'm Amparo Galindo, and I'm the chair of the institute. Okay, so with that, let me welcome Karen Sarkisyan. He is the co-founder of Light Bio and head of the synthetic biology group here at Imperial in the Faculty of Medicine. Again, you know, this demonstrates the interdisciplinarity of the work that we do. And Karen, it's a pleasure to have you here. I will let you run through your title and your talk. Thank you.
Thank you very much. Thanks, Sloan, for inviting me. My talk today is about genetically encoded bioluminescence. And actually, is it possible to dim the lights a little bit, because most of the slides are dark? So I will start with a clip from the Avatar film. Yeah, perfect. Thank you. So this is a clip from the Avatar film, which is essentially, I think, the largest budget movie about bioluminescence. And in the Avatar, there is this planet Pandora, where there is an amazing ecosystem which is full of bioluminescence. You can see bioluminescent plants, bioluminescent fungi there, bioluminescent animals. Plays a big role in communication between the organisms. You can see examples of light-based computation in that ecosystem. And if we're doing engineering biology, the question is, can we do this kind of stuff here on Earth? Can we engineer organisms to glow in the dark? The real answer is not yet. We don't have a good technology yet, but we're getting there. And essentially my talk today is an update on how this technology has developed over the last years and where we are in terms of our ability to engineer bioluminescence. So we also have quite a lot of bioluminescence, of course, on Earth. And different organisms, they glow for different biochemical reasons. They all make some molecule which, when oxidized by air, emits light. But in between the thousands of species that we have that glow in the dark, there is about 40 different, biochemically different and evolutionarily different bioluminescence systems. So they all make different molecules, but all those molecules are substrates for this final bioluminescence reaction. And if we want to engineer new organisms to glow in the dark, we need to understand the biosynthetic pathways that lead to production of that molecule in the cell from some core cellular metabolites. So the key question is how to produce the substrate for luminescence in the new organism. And surprisingly, out of there's thousands of species that we have, we only understand enough, just 2 pathways. One comes from bacteria. This is the one on the left. It's actually been known for about 40 years now. It's encoded in what's called lax operon, 6 genes clustered together in the bacterial genome. And it's essentially, biochemically, it's a branch of fatty acid metabolism. The other one was discovered more recently. It's the pathway from some tropical fungi. There's about 100 species of fungi, mostly in the tropical forests, and they encode also some kind of a genomic cluster with 4 genes which encode 4 enzymes that catalyze this pathway. And this pathway, biochemically, is a part of phenylpropanoid metabolism. We work on both pathway engineering of both pathways, but this work has gone a bit further, and so I will mostly talk about the application and engineering of the fungal bioluminescence pathway. You can see the chemistry of the pathway here at the center. It's called caffeic acid cycle. It starts with caffeic acid, which is converted to this compound called hispidin in a series of reactions, all catalyzed by one huge enzyme called hispidin synthase, HISP-S here. Hispidin is then oxidized to produce that molecule that actually gives light, so the substrate of luminescence reaction called free hydroxyhispidin. It's oxidized by luciferase enzyme to emit light, to produce oxyluciferin, which is then very nicely recycled back into caffeic acid. And so it's a cycle that essentially converts the energy of the plant into light. And immediately when we saw the chemistry of it, we thought, it's almost plant metabolism if you look at it. So here on the slide, you see plant metabolism in white and fungal pathway in blue. And you can see there is a lot of overlap already. So the idea was maybe we could just overexpress a few genes, and that will be enough to make plants glow in the dark. And that's exactly what happened. So we overexpressed a few genes in tobacco plants, and we got plants that were glowing continuously in the dark, bright enough to be visible to a naked eye with some eye adaptation. And essentially, they were glowing throughout their lifespan, from germination to flowering. And that was very, very encouraging. And also the fact that we could image those plants not on this high-end luminescence imaging equipment, which costs usually hundreds of thousands of pounds, but we could just use normal cameras, which are like 100-fold cheaper to image that. Which means that in the lab, you don't have to invest so much in your imaging experiment. You can buy a lot of those cheap cameras and scale it and run many experiments in parallel. So we're thinking maybe that's a good system to actually create a platform technology for all kinds of imaging tasks to image molecular physiology at the level of the whole organism. And it's especially important for plants because in plants you cannot really use fluorescence-based approaches, which are very good nowadays and very popular. But in plants, as you can see here, this is a plant under UV light. It's all autofluorescent. It's very autofluorescent. It's packed with pigments. If you overexpress your GFP on top of it, you don't get such a good signal-to-noise ratio. With luminescence, it's different because unless you express your luciferase gene, your organism would typically emit 0 photons, so there is no background. And with the luminescence system, which is fully genetically encoded in a plant, we thought there could be very different interesting applications. For example, we could potentially monitor a spread of infections in real time. You can imagine a plant that produces the substrate, but then the last enzyme is delivered with a virus, and then you image the spread of viral infection in the plant. Or you can think of plants as sentinel organisms which light up when they sense something in the environment. Or actually, because you don't need to provide substrate externally, the substrate is produced intracellularly, you can actually move away from the lab and start imaging physiology in the soil, in the real complex ecological environment which hopefully gives us much more relevant information about plant physiology and plant responses. But at the time, if we just had the wild-type fungal pathway, the technology was not robust enough, was not good enough to just be applied to all those stacks. So a few years ago, we kind of made a list of things to do before it can be actually useful. One thing was to actually make it work in plants which are different from tobacco, because the wild-type fungal pathway didn't work that well. The whole system was quite big, and so we had to compress it, and we want to compress it, we want to make it very user-friendly. And the other big problem with all those approaches is that if you're interested in understanding the biology of a plant, say, those systems actually don't give you a very quantitative signal. The bioluminescence you're getting back is not just a reflection of gene expression, which you would typically want from your reporter tool. It's also a reflection of activity of cell metabolism. So if a cell is very metabolically active, you would get a lot of substrate, a lot of signal at the same expression level of genes. If it's not, you'll get very dim signal, and then the whole thing becomes very non-quantitative. And so we're thinking about ways of how to fix this. And finally, the fourth thing on the list was to create those cheap and easy-to-use imaging devices for plant physiology imaging. And so on the first point, there's some pathway engineering work and enzyme engineering work led to essentially improved version of this bioluminescence pathway, fungal bioluminescence pathway version 3, we call it. We combined, we screened different enzymes from different fungi, found the more active ones, applied directed evolution with our collaborators to improve some of the genes, improve their thermal stability and activity, and also figured that we need to express the fifth gene to make it broadly applicable across the plants. And that resulted in this pathway FVP3, which is now, we're confident in saying that it's a good enough technology to actually use. You can apply it to Arabidopsis, works quite well, which is a relatively distant plant from tobacco. You can apply it to chrysanthemum, to actinidia, to poplar as an example of a tree. So it seems to be working quite well in a broad range of plants. And it seems to outperform all the previous versions of the system. We also figured how to make this huge enzyme, hispidin synthase, smaller by replacing it with a much smaller enzyme from a plant origin, which seems to be doing roughly as well, a bit worse than the fungal enzyme, but still quite well. And so that allows you to really compress this large gene and encode the thing in a virus, if you're interested in imaging viral infections. And on the commercial side of things, the company called Light Bio this year brought this plant, Petunia plant, to the market. This Petunia expresses not even FVP3, it expresses FVP2. And it's a bright enough plant to be a commercially viable product. Here you can see photos. This is the production facility of Light Bio. I think it's a good example of what you can expect in the field, right? If you want to create lots of bioluminescent plants and image their physiology in the wild, image their interaction with pests, with pathogens, this is the kind of field you want to have to be able to see the spread of, stress response across the field or something like that. Some photos from social networks. But the point I would like to make is that it seems that this technology is bright enough that it allows not only imaging plant physiology at the time scale of the whole lifespan of a plant, it also allows you video rate imaging of very quick events in plant physiology. So for example, here's an example of that. This is the reaction, phenylpropanoid activation upon wounding of the plant. You can see quite a quick response of the plant to the wounding. Here is another example. where we use the bioluminescence system as a reporter tool for hormone activity. So salicylic acid is a hormone involved in plant stress response to pathogens. And you can see that if you add salicylic acid to the plant that conditionally expresses the bioluminescence pathway, you get great activation of light emission. So one of the big questions is how to actually make this whole thing quantitative. And as I said, the signal we're getting is not, unfortunately, just the gene expression we're interested in. It's also a function of metabolism. And so the question was how to figure the way to make it, to disentangle gene expression and metabolism in the signal. And we thought we could, this is an illustration of how the metabolism changes light output over time. And the solution we're thinking of is essentially, what if we could have two colors of luminescence? Identical enzymes, identical catalytic properties, but two different colors, and we just co-express them in the same cell at the same time. And if one enzyme was expressed under a reference promoter, and the other enzyme is controlled by a different promoter, the one you're interested in studying, then the ratio of two colors, and essentially the final color of luminescence, will reflect the ratio of activity of those promoters, but because the enzymes are using the very same substrate, it will be independent of metabolism. So it's kind of a ratiometric sensor that allows to disentangle gene expression from metabolism. And as a result, so we're kind of halfway there, not finally there, but it seems to be working. And as a result, you get color change, not light intensity, but color change as a function of whatever you're interested in. And that makes it much more quantitative. Finally, we've also been working on making this an easy-to-use imaging tool. We're designing those rhizodrones where you can grow plants with lots of cameras around them and see both shoot physiology and root physiology at the same time, with using these cheap consumer cameras. This is an example. Reminds me of how our streets look nowadays. Essentially, those poor Arabidopsis plants, they grow in this thin layer of soil, which allows you to image the roots quite well. And also the shoot you can image from different angles. I think that's it for me today. That's an update. I think we are kind of halfway towards being able to engineer things like you see in the Avatar movie. I think hopefully in the next few years, the technology will be mature enough to engineer much more of that. Thank you. And yeah, just want to say that this is, of course, a hugely collaborative work with a major part played by two companies, Planta and Light Bio. And some other groups, our group at the LMS, Yampolsky Group, Mission Group, Diego Arzai's Group, Key Food, Promega Advanced Technology Group, Kondrashov Group, and Petrashek Group. Thank you very much.
Thank you, Karen. You can stay here for questions. Yeah, go ahead. Do you need? I think if you can wait for a microphone. Or just, yeah, if you can pass him down. Thank you.
Thank you. The way I'm seeing what your setup is at the moment for reporting on these things, it's going to be expression of your actual proteins, which is slower than the signal might actually be there. So have you looked into making hybrid proteins with an actual detector domain, and whether that could then detect whatever your stimulus is or a small molecule, and then activate the already present protein, whichever one in your pathway?
Yes, that's a great point. So it's exactly the problem that, it's transcriptional-based reporters so far. We have tried making some non-transcriptional-based reporters, failed. We still want to do that. Part of the problem is that luciferase is a membrane protein, so it's a bit harder to engineer. Also, you don't have access to all the metabolites next to the membrane. Some of them may be compartmentalized in the cell, so that's part of the problem. But yeah, I think that should be possible. Thank you.
Thank you very much. It's a really inspiring talk. So I was wondering, so obviously you've got a challenge about the amount of essentially energy input into the pathway to give you the light, but is there anything in the chemistry, because you're generating excited electronic states, whether there's any sort of free radicals or reactive oxygen species, and is this a challenge that needs advancing?
Mm-hmm. So I don't have a good answer. I think, so we looked at the phenotype of the plants, and with this FBP3, we couldn't detect any difference in phenotype. In the original system, we did detect some differences. For example, plants that expressing the system were a bit taller for some reason. We don't fully understand why. But we didn't see any significant kind of burden yet. So maybe we're not at the right level of light emission to see a significant burden. I think those reactions are pretty standard for plant metabolism in general, like phenylpropanoid metabolism. So I would imagine plants already know how to manage those things, unless it's expressed at the huge levels.
Thank you.
Thank you.
Yeah, really cool talk, thank you. You mentioned in the avatar clip something about computation, and I guess I was wondering, is there a pathway to communication with light? So here you're generating the signal, and then with cameras we can measure things, but could organisms communicate with light?
Yeah, thank you. So if you watch the film, you know that they used to connect their, whatever it's called, the organ they had here in the hair, right, to the tree, and the tree was bioluminescent, and the thing was bioluminescent. And then I don't have a picture, unfortunately, but essentially, I think it's some kind of bioluminescence optogenetic system that was used to communicate with trees and between animals and trees. So you can imagine like optogenetics in the nervous cells, bioluminescence exciting optogenetics. And there are works now that show that you can excite optogenetic channels with bioluminescence. Just not autonomous bioluminescence yet, but maybe it's just it's going to happen in the next few years. So it's possible, it should be possible to engineer communication at the cellular level. It's not done yet for autonomous systems. Yeah. I don't know if I answered.
Yeah. Yeah, it's something that we've thought about, and I guess struggled with the coupling at the moment between luminescent proteins and optogenetic proteins. So I know it's a difficult
We also
thing.
had a project like I think 7-year project now, which is still unpublished, which was trying to do something like that. Yeah, so it is difficult.
Karen, I think I might have asked you this one before, so apologies if you've already told me. Fungi is where you got the bioluminescence from, is that right?
Yeah.
And then obviously we know lots of animals that do bioluminescence. Why do you think plants don't naturally do this?
Yeah, that's a good one. I think it doesn't make sense for them, most likely, because I think plants have all the chemistry, all the capabilities to be bioluminescent if they wanted to, if there was a reason to evolve it, and including the precursor of the fungal luciferin. So some plants just naturally produce it. I think as a way to attract pollinators doesn't make much sense because it's very energy intensive, and also you end up with only maybe 3-4 colors as a channel to communication. Whereas what we see in evolution of plant pollinator interactions is that there is a lot of specialization, right? You want a molecule that can be sensed 1 kilometer away from your plant that will attract your specific pollinator and bring it to the plant of your species, right? And that, I think bioluminescence just doesn't allow that to do that because you are limited to 3-4 colors.
Any further questions? No. Do we have any questions online? Leah? No. Further questions? No? Okay. Join me in thanking Karen for a really inspiring talk. Okay, and with this we welcome our next speaker. Is Miguel Paez Perez. He is a research fellow in the Department of Chemistry here at Imperial. Thank you for being here, Miguel. And I give you the floor. Yeah, the clicker. This is a clicker. So this is that's your point there. Yeah, that's your point there. Yeah. Okay, thank you.
Hello. Yeah, I guess this works. So perfect. So thank you very much, Amparo, for the introduction, and hello everyone. So my name is Miguel, and the topic of my talk today is how we can benchmark optical probes. And this actually relates quite well to what Karen was pointing out before regarding the need to perform quantitative measurements if we really want to understand what's going on in our system. So in particular, we are interested in soft interfaces in lipid membranes. And the reason of being interested in these assemblies is that lipid membranes are quite important in biology. And for example, their biophysical properties will be a biomarker of disease or the metabolism going on in different organisms. So some of you might be aware that one of the gold standards to measure the biophysical properties of lipid membranes is the use of environmentally sensitive molecular probes. And you can see here that there's a wide range of them with different advantages or probing different parts of the membrane, like the head group, the tail, and different physical properties, for example, the charge, viscosity, and so on. So in particular today, I will focus on a family of dyes named molecular rotors. And these dyes have the advantage of being sensitive to viscosity. That is the microenvironmental molecular crowding. So the way they work is shown here. So basically, you have this molecule which has two different parts, and there's an intramolecular motion between these. Now, the degree of this rotation or of this motion is dictated by the molecular crowding around our probe. And therefore, in a lower viscosity environment, where there is less crowding, the molecule will rotate faster, and this will be reflected in a lower intensity of the fluorescence and also in a lower lifetime. And the opposite will happen in a higher viscous environment. You will have higher fluorescence intensity and higher fluorescence lifetime. And very importantly, when we design these probes, we want them to be independent of other confounding factors, such as, for example, temperature, polarity, and so on. So the way we start our experiments, let's say, is by calibrating the response of these probes to viscosity. And we do it, as you can see here in this plot, so we can measure the macro viscosity of a solution and then measure the response of our probes. In our case, we use fluorescence lifetime because it's independent of concentration, and that already takes out one confounding factor when trying to aim for quantitative results. But nevertheless, what you can see here is that for a given viscosity, then we can get multiple fluorescence lifetimes. And the reason is that the degree of molecular crowding, that is microviscosity, is ill-defined usually in bulk solutions. And as you can see here, for example, the response that we get with glycerol is not the same one as we get with a higher molecular weight polymers such as PEG. So this is actually quite relevant when we study lipid membranes, and the reason is that these structures are incredibly complex and highly varied. And even if we remove the proteins, which are an important component of them, then we end up with a lipid bilayer, which itself is highly heterogeneous. So as you can see here, we have different features. For example, we can have a phase separation between different lipid domains. We can also get regions of higher curvature, and we can also as well get asymmetry between the two membrane leaflets. And all of this is introducing confounding factors when trying to interpret our results. So here comes the need to benchmark these fluorescent probes in terms of the actual structure of the membrane instead of some calibration that we can do in the bulk in a system which doesn't represent at all the biophysical properties of this bilayer. And we do so by comparing structural data that we obtain from X-ray diffraction For example, we can use small angle X-ray scattering to get information about the thickness of the membrane, and we can use wide angle X-ray scattering to get information about the average area that each lipid is occupying within the membrane. And we compare these results to viscosity or lifetime values that we get from our molecular rotors. For example, here a body dye that sits in the region of the hydrocarbon core or the tail of the lipids. So we can then slowly change the temperature, and this allows us to gradually vary both the structural properties and the kind of viscosity of the membrane. And as you can see here, we can get some kind of simple relationships between the structural properties and the viscosity of the membrane as we change temperature. So, of course, this then allows us to establish an analytical relationship between the membrane viscosity that we can measure through optical methods and the structural organization which we measure through X-ray, which, of course, is not biocompatible, for example, and it will not allow us to obtain spatial information of the membrane. And as you can see here on this plot, here on the bottom, we can then calibrate the response using some model lipid, for example, the OPC here. And as you can see, the response of other lipids in the same fluid phase will be the same. And therefore, this allows us to use optical imaging in order to infer structural parameters. And here you can see one example. So say we have phase-separated membranes, as this one here on top, which are representative of those in cells. If we look at the X-ray pattern, then we cannot really distinguish the two domains. However, these are very clearly seen in film, which is a microscopy technique used to measure the lifetime of the probe. And then if we do the Carrier transformation of the viscosity values, we can then infer the area per lipid for both phases, which actually matches quite well with molecular dynamic simulations, as well as those obtained from pure X-ray diffraction experiments. And this approach also works quite well with biologically relevant lipids. For example, here you can see that we can use our method in order to infer the area per lipid of E. coli lipid extract. So this altogether gives us confidence on our method. So now I will just try to show you another example of how we can use the combined X-ray diffraction and molecular rotors approach in order to reveal non-classical behaviors of lipid membranes. So you can think of a membrane as a rubber band, and therefore, whenever you stretch it, for example, using hyposmotic shocks, then the area per lipid will increase, and therefore the thickness will decrease, as well as the order and viscosity of the lipids will be lower as well. And the opposite will happen when we compress the lipids together. So I will focus, for example, in the hyperosmotic case now, where we press the lipids together. And what we can see is that DOPC, again, this is a lipid in the fluid phase, so pretty much our ideal rubber band. When we squeeze the membrane together, we see an increase in viscosity, which, again, makes sense because we are pushing all the lipid molecules together. However, when we have lipids in the gel phase, fully saturated, this means that the lipid molecules are very closely packed together and they cannot compress anymore. If we still apply some pressure, what we see is a decrease in viscosity, which was surprising because this kind of means that the membrane is becoming more disordered as we squeeze it together. So we then decided to do X-ray diffraction experiments, and what we see is that the area per lipid decreases. So we are kind of actually squeezing the lipids together, but still we are getting some disorder. And the reason for this, we can see it here in the sacs, where we study the difference in membrane thickness. So for the rubber band case, the DOPC case, we see that the thickness increases, which makes sense. We squeeze something together, then it becomes thicker. However, in the case of DPPC, where the membranes are highly ordered, the thickness goes down. And we believe the reason for this is the existence of this kind of highly curved membranes, which create pockets of regional disorder. And this is actually quite relevant in biology because this process, which was not experimentally observed before, is thought to play a role in the hearing process happening in the cochlea. So this is just one example, but with this, I just wanted to give you the following take-home messages, hopefully. First, that we can use optical probes, in particular molecular rotors, to quantify the biophysical properties of soft interfaces with spatiotemporal resolution. But, of course, we cannot rely only on the readout from these optical dyes. We need to use alternative techniques, for example, X-ray diffraction, in order to benchmark the response of these probes. And this will allow us to get a unique insight into the very complex and non-classical behavior of lipid membranes. So with that, I will like to acknowledge the Marina's Pumaba's lab, the Membrane Biophysics group, and the Miguel's lab, and our collaborators and funders. And of course, thank you for listening.
Thank you, Miguel. Very nice talk. Thank you for a very nice seminar. Any questions? I'll start with one while they think. Is there any role there must be, or any works doing molecular simulation of these systems?
Yes, so we have done MD simulations. Okay. I mean, we and other groups as well. Mm-hmm. And you can actually see that the dyes in this case sit at different regions of the membrane, which again brings us to the problem of like we cannot really know from bulk calibrations what's the environment that the dye is sitting in. And yeah, in general, like for every measurement that we do, we need to do MD simulations or like some other kind of theoretical approach in order to have an insight on whether the probe relocation will influence and to what extent the readout from these probes.
Okay, perfect. Yeah, because it seems like you've been going from, like you're showing, from the molecular arrangement to the properties that you measure.
Yeah. So that's a big step.
Yeah. Fantastic. Okay. Any other? Karen. We need a microphone.
Thank you. Could you see these probes as having a role in studying membrane dynamics in, say, osmotic shock conditions in single-celled organisms, in bacteria, for instance?
Yeah.
Or is it potentially too complex a membrane system?
So it will be more complex to interpret the results, and that's the reason why we are trying to follow this approach. But indeed, we already have done some experiments where we use these dyes to stain the membrane from E. coli, and you can measure the effect of the different stressors on the membrane of bacteria and also from some other eukaryotic cells. So yeah, that's something that is quite established. Thank you.
Thank you.
Thank you. And we have a question here. Thank you.
Thank you. Very interesting. I'm not sure, maybe I missed it. I'm wondering what's the kind of time resolution that you can achieve with this imaging, and how does it, correspond to the actual time scale of the molecular viscosity changes that you can expect from living systems?
So in terms of time resolution, it depends on the modality that you are using. So there's some families of ratiometric molecular rotors. In that case, you can do in the millisecond range. If you are using the lifetime, then you are basically limited by the accumulation time that you need in order to build enough photon count to get a proper fit. As a rule of thumb, you can go, if you are, you're like in an optimized system, let's say, you can go like 10 seconds. Mm-hmm. Ideally, half a minute is a good kind of time resolution that you can get. But these processes usually take around 10 minutes to kind of stabilize. So I think you can keep some, it will not be like super high time resolution because again, you are limited with this, but you can measure the kinetics.
Thank you.
You're welcome.
Do you have any other questions? Online? No. Okay, then right. Let's join me in thanking Miguel again, please.
Thank you.
Thank you. And I can keep this one. And our next speaker is Kyler Roy. He's a PhD student in the Department of Bioengineering in the RLA lab. Thank you for being here, Kyler.
Thank you.
So, over to you.
Awesome. How's everyone doing? Good. Nice. As mentioned, my name is Kyler Roy. I am a PhD candidate in the Rodrigo's group in the RLA lab. And today I'll be talking to you about my non-repetitive CRISPR AI toolkit for mostly for metabolic engineering purposes in Saccharomyces, whatever you say. Do we have a lot of wet lab people in the building today? Yeah, show of hands, everyone? Sweet, that's like most of us here. So I want to start off this presentation about everyone thinking about a really annoying task that's repetitive that you do day to day in the lab. It could be mini preps, glycerol stock labeling tubes, whatever you want. I want you to think about why you hate doing this particular repetitive task. And for me, it is qPCR. And the reason why I hate doing qPCR so much is because I routinely mess it up all the time. And so we'll have this nice qPCR plate over here. I'll be adding in reagents in well 1, 2, 3, and eventually I'll get to somewhere like here. And I'm like, did I add the primer? Did I add the template? What should I do next, you know? Should I assume that I added it and I potentially double the concentration? Should I assume that I did not add it and potentially miss it? Should I redo the whole plate and just waste all these reagents? Right, so show of hands if something like this has happened to you in the lab. Pretty much everyone. And if you did not raise your hands, you're either a robot or a liar, because it is literally in our DNA to make these repetitive mistakes. So even one of our favorite enzymes, DNA polymerase, as it's going through repetitive streaks of DNA, it can either duplicate these regions, delete these regions, or slow down and take its time to process it properly. So it's literally in our genetics to make mistakes in repetitive tasks. And you're probably thinking to yourself, why do I care? This isn't a therapy session for us all in the lab, why we make mistakes. We care because we're bioengineers. And one of our favorite bioengineering tools is CRISPR. And CRISPR is inherently repetitive. It's literally in the title, right? And while there are many CRISPR tools available that people use for engineering, today I'm mostly going to talk to you about CRISPR activation and inhibition, which consists of using a dCas9 fused with an activation domain or a repression domain, which could target the Cas proteins to a promoter of region, which can activate or repress genes as necessary. And the beautiful thing is this entire process can be multiplexed. So by using one single transcript, you can express many guide RNAs simultaneously to activate or repress as many genes as you would like. And by using orthogonal Cas nucleases, such as dCas9 over here on the left and dCas12 on the right, fused to an activation or repression domain, they recognize their own specific guide RNAs, which again, down below, have distinct structures. And by recognizing these distinct guide RNAs, you can essentially activate or repress genes simultaneously with zero crosstalk within our system. And this is an amazing tool for metabolic engineering, which is essentially trying to produce a particular metabolite of interest in greater quantities. So I apologize for this disgusting metabolic network, but for example here, if you want to make succinic acid down below, you can simultaneously activate and repress many genes along this network to shift flux right from your feed to your metabolite, increasing your yield. And while our system has some easier components to express, like the Cas and the nucleases up here, expressing the large polycistronic multiplex array has been much more challenging to do. And the reason why it has been much more challenging to do is due to its repetition. And to kind of explain that, I need to go through the anatomy of a multiplexed array. So starting on the left here, we have the guide RNA protospacer. So this is the base pair target that goes to, that targets the Cas and the nucleases to the site of the genome of interest. Next, you have the Cas9 scaffold. So this is part of the guide RNA that allows the Cas enzyme to recognize it, grab onto it, hold onto it to get delivered to the genome. Next, you kind of have a bacterial terminator or a Cas9 terminator that kind of signals the end of the guide, so adds stability and ends transcription. followed by a CSY4 recognition site. So CSY4 is kind of like a molecular scissors. It recognizes these little hairpins and able to cut at these sites to liberate all the individual guides along the array. And then finally you have the Cas12 scaffold, which again, like the Cas9 scaffold, is just recognition sequence by the Cas enzyme to grab onto that guide RNA. But the main thing I want to show on this slide is that these 4 circles here are all appear along the length of the guide RNA array, and they're all repetitive DNA. Right? To kind of simplify this a little bit more, you'll see you have your protospacer scaffold CSY4, and this light yellow and blue happens all the way throughout the multiplex array, and this can be to the end. It's all repetitive DNA, and in a current system, this can be up to 24 times. And because of all this repetition, the commercial synthesis of this array is unavailable. It's hard to do. So that makes, so for example, a couple years ago, we released this manuscript where we made a multiplex array to target 24 genes. And as a proof of concept, we made this 11 guide array to activate these 2 genes in green and repress these 9 genes in red in central metabolism of Saccharomyces cerevisiae to increase the yield of succinic acid. So if you take this 11 guide array down here, and for example, if you want to punch it into IDT and order it as a gene block, you'll immediately get this error saying, no, it's impossible. It gives you a complexity score of 454. And this is largely due to all the repeats in the sequence. So you just can't order it as a G block. All these repetitive sequences make it hard to commercially synthesize. So that means you have to clone it the hard way. And so by PCR amplifying and Golden Gate cloning, you can assemble these large arrays, but it's incredibly inefficient. And 9 times out of 10, I get plates that look like this. Absolutely 0 colonies. And at first, I kind of struggled, and I was like, oh, maybe I'm really terrible at cloning, but it's just kind of the nature of the beast of when you add repetitive DNA, it's just low success rate. But then every once in a while, you do get some success. You do find a couple colonies on your plate, and then you pick them up, you send it for sequencing, and you see this large error. And this isn't just a part being missed, it's some sort of recombination event. So months and months of just empty plates, terrible sequencing, it just made me really stressed out. My hairline kind of went up like at least like 5 centimeters during my entire PhD. And it's been challenging. So you might be thinking to yourself now, but like when you do have this plasmid, it must be stable forever, right? And you're absolutely wrong. So if you were to take a 24-guide multiplex array, for example, and you serial passage it over several generations and send it for sequencing after so many days, you'll notice that over time that you get more and more and more deletions and duplications. And to the point where after 7 days, you almost see your entire array knocked out in E. coli. And this is consistent with the literature. So there is this EFM calculator. So it essentially measures the instability of plasmid DNA in E. coli based on repetitive motifs. And if you take this 11-guide array I mentioned earlier, you punch it into this calculator, it gives you an instability score of 29,000. So that means it is 29,000 more times likely to mutate per generation than the baseline E. coli. So now you might be thinking to yourself, but what if I just transform this plasmid into your host yeast really, really fast? You know, it has to be stable at that point, right? If you genome integrate it into Saccharomyces. And you're wrong. So to kind of test this, I made a BFP reporter assay, essentially controlling BFP and targeting it with either a single guide, 3 guides, or 6 guides. So essentially 0 guides, 0 repeats, 3 repeats, and 6 repeats to repress the expression of the blue fluorescence protein. And so when you look at the flow cytometry data, where we have different passages here on the y-axis and the fluorescence intensity on the x-axis, you can see at day 0, you see a large amount of blue fluorescence. Then once you target it with the guide RNA, you see this nice leftward shift. And when you have a single guide or 0 repeats, this decrease in fluorescence is stable across all 7 days. But when you add 3 guides or 3 different repeats, you'll notice that approximately on day 4, you'll see this subpopulation emerge. And it's reverting, you see this rightward shift, and it's reverting back to its baseline. And then over time, this baseline population of BFP becomes the most dominant and the only population remaining. And more interestingly, when you have 6 guides targeting it, at day 4, you see this escape. It's much, much, much more prominent than in the 3 guides. And this shift is much more dramatic at day 5 as well, where it's the only population remaining. So just to kind of summarize, is more guides equals more problems and more instability within the multiplexed array. And this is confirmed by colony PCR, where you can see where I amplified the region flanking the multiplexed array. And at day 0, you can see a nice little ladder here between 1 guide, 3 guides, and 6 guides, where at day 7, it's almost completely gone. So you can see that it's not the CRISPR proteins or the BFP getting mutated or lost in the array. It is the array, in the system, it's the array itself that is recombining and getting lost over time. Right, and this is obviously a massive problem for metabolic engineering. So again, if we have the system that makes succinic acid and all the flux shifting from feed to metabolite, and you slowly start losing your guides to alter this metabolism, you eventually start losing your yield, your titers, and eventually you go back to your baseline strain that is no better at producing your metabolite than the wild type. Right, so how I'm fixing this is that, again, CRISPR stands for clustered regularly interspaced short palindromic repeats. It has all this repetitive DNA. If you remove all this repetition and make it unique sequences, essentially you can become CRISP, where you get rid of all this repeats. And to do this, I took all the 4 repetitive parts of our multiplex array. I mutated the individual bases while maintaining the secondary structure. And if you do this, the Cas enzymes can still recognize these distinct hairpins and use it as a functional guide. I then took all this mutant library and used a flow assay to kind of detect their ability to decrease GFP fluorescence. And then took these individual CRISPR elements, fused them together as one distinct self-processing CRISPR AI unit. And then I add them all together on one single plasmid to make the non-repetitive CRISPR toolkit. So you might be asking yourself, are the non-repetitive guides any better? And earlier I showed that you can't commercially synthesize a repetitive multiplex array. But if you take my system and you utilize or try to order as a G block, it gives you a complexity score of 1.9. So using non-repetitive guides, you can order the exact same multiplex array as a G block, so easier to clone. Earlier I mentioned that the repetitive guide RNAs are unstable in E. coli and have an instability score of 30,000 approximately. Using the non-repetitive guides, you get an instability score of 1.2, so just marginally more unstable than the baseline E. coli. So are they better for metabolic engineering? And this is what I'm currently working on in the final phase of this project. And to do this, I'm going to make raspberry ketone. And so it is a nice fragrance used in the perfume industry, as well as a nutraceutical for weight loss. And to kind of test this, I'm going to design a 24-guide repetitive array and using the exact same guides, but non-repetitive versions, just measure raspberry ketone in a bioreactor over 7 days. And hopefully, the non-repetitive arrays become more stable and increase the yield over this time period. So just a quick summary is repetitive DNA is bad. You know, it's hard to work with. It's annoying, low efficiency. It's unstable. If you can avoid using repetitive DNA, I highly recommend you do that. And that non-repetitive guide RNAs, to be determined if they're better, but theoretically at this point, they seem to be a lot more easier system to work with. So obviously I'd like to acknowledge everyone in the RLA lab, as well as my supervisor Rodrigo, for the opportunity to work in the lab and to learn, as well as the entire Center for Synthetic Biology at Imperial, and obviously all the sources of funding that allowed this to happen. So as of now, that's the end of my presentation. If anyone has any questions, feel free to ask right now. And if you think of one later, don't hesitate to email me at kbr20@ic.ac.uk. Hopefully I can get back to you shortly with that. And that ends my talk. If anyone has any questions, Giver.
Thank you, Kyler. Really nice. Resonated at so many levels. Yeah. I do computation, so but repetitive tasks, even in computation, are a pain. You know, eventually your brain switches off. Amazing that you should. We have time for questions. Any questions? Yes, Cristala. Yeah.
So first, thank you for your honesty. It's useful, I think, for people to hear what's really going on. I have two questions, one of which you may have kind of answered because you said it's yet to be seen if it's any better.
Yeah.
And you have a specific application, but I'm wondering if you've tested it even on simple reporter proteins to see if making those changes have any impact on the degree of activation or repression that you see.
So all the different versions of these guide RNAs do have different activating and repression levels. The ones I selected for the toolkit will have individually have upwards of 90% effectiveness of the wild type. I am currently doing the flow experiment again with the non-repetitive guides to see if it's more stable in that regard. I don't have the data for that at the moment though, but I'm currently working on that assay as well, or that reporter.
And so then my second question is, do you have a sense as to what the upper limit is, right? You referenced a previously published paper that had 12 arrays, difficult to produce. Any sense of how much, how many you can make and what the upper limit is on that? Is it the ability to generate diversity so that you minimize repetition? Is it the size of the cassette? What are the factors that will determine how many of these you could do at once?
I mean, all those things are obviously coming into play. In our manuscript that I mentioned earlier in the presentation, we found that the guide RNAs actually express themselves even without a promoter. And if you go beyond 24, you start seeing a lot of leaky expression of the guides. So depending on what type of CRISPR application you're using, you can start creating a lot of burden. And so it was a lot of work on this old postdoc, Will Shaw, that he made to really silence the array when you don't want it to be expressed. So the problem once they get bigger and bigger, at least in Saccharomyces, is that you'll have this leaky expression, and the more guides you have, the more burden you'll have. So it all probably depends on the context of how you want to manipulate metabolism.
Great, thank you.
Thank you very much for the talk. I'm sure you already tried this or something similar, but I'm still curious to know what is the improvement if you use some strain of E. coli or Saccharomyces that is simply defective for recombination?
So the E. coli experiment that I showed there is actually an Arec negative strain, so it's still recombinated or had recombination happening. I'm currently working on trying to investigate the mechanism of the knockout of the array, and so there's many different ways this repetitive sequence can be removed from the cell. One is homologous recombination. Another one is genetic slippage. So the slide I showed where DNA polymerase is stalling doing repetitive stretches, essentially what can happen is it can make a hairpin loop and then stall and then delete that entire array. So it has nothing to do with recombination, it just has to do more with the replication machinery just emitting that repetitive stretch. So I'm trying to figure out exactly the mechanism behind it. So for E. coli, using a Rec A negative strain, it doesn't help. And in Saccharomyces, one of the beautiful things about the organism is the ability for homologous recombination. And that allows you to engineer the strain for almost whatever purpose you want. Easy to work with. And so if you start knocking out this recombination ability, you're making it much more harder to engineer for your application. So there's probably some sort of trade-off between it. Maybe using an inducible CRISPR to knock out homologous recombination after you engineered it. I don't know, there's some strategies around it I'm thinking about doing, but at least in E. coli Rec A negative strains don't seem to help, and engineering Saccharomyces to be Rec A negative essentially might be detrimental for engineering.
Thank you.
Hello, thank you for the great talk. Do you know if the wild type CRISPR has has the same instability, or does it have any mechanism to keep it there?
So when we engineer our CRISPR systems, we use a single guide RNA. So there's different guides in the native bacterial system that they'll fuse together as one. In engineering, we fuse it together as one single transcript, essentially, which adds to repetition and makes it more simple for engineering, but more repetitive in this regard. In native bacterial systems, though, it is a little bit more stable because you don't necessarily have the single engineered repetitive sequence, but they found over time that when you have this array all the guides are added sequentially along the array, and that's kind of like a molecular recorder of past infections. And so they found that the arrays at the ends are a lot more stable than ones in the middle. So again, this kind of leads to that it's more of a genetic slippage event causing the arrays to get kicked out over time. So they do see it's unstable, probably a little bit more stable than what I'm seeing in the lab, but because of its, molecular advantage to have these guides for defense, they probably act as an advantage to keep this array in there, and they find that more recent and more distant infections are a lot more stable than the ones in the middle of the array.
Thank you.
Okay, thank you. I want to ask, how about the future direction of that project? I mean, maybe like trying to try different kind of Cas9s or the variant of Cas9, and then how about the fitness of the array and the Cas9s? Yeah.
Sorry, I didn't quite.
Yeah. The future direction of your project. You can you try different variant of Cas proteins for that kind of. Yeah.
I mean, that'd be an interesting way to do it because each Cas ortholog has a slightly different guide RNA structure, but then you need to express many different Cas enzymes, which are obviously burdensome to a cell. So if you want to have many guides targeting the genome simultaneously, it's probably advantageous to reduce the amount of cellular burden and reduce the amount of Cas orthologs in there. So I mean, it could be a strategy for sure using different orthologs, but then you're adding a lot of protein burden, and so I don't know if it'll necessarily be advantageous to do that.
Okay.
Any further questions? Yeah, I'll give you the microphone.
Great talk. I was just wondering, you use Cas12 as well as Cas9. Do you have any trans cleavage issues with Cas12?
So we use a dead Cas for both, so we don't see any cleavage activity.
Got it.
Yeah.
Thank you.
Just kind of following on that, does the transcleavage activity, I thought the transcleavage activity happens because of a fold change and then an opening of a kind of a nucleus site. So can you make that dead, is the dead Cas dead in both cis and transcleavage modes?
I haven't noticed any. I believe that they're both nuclease activity is dead in these.
Fair enough.
Yeah.
Yeah. I'll look at it.
Yeah, thank you. It was a really cool talk. With the RNA, designing the new versions, I guess I'm just interested to hear how you did that. There's lots of literature coming out on machine learning ways to do it, and I guess there's other ways.
Yeah.
And what actually works?
So there's a couple other groups that done very similar stuff in E. coli. So some of the guides I stole from these other papers. Basically, they use some sort of machine learning or some sort of, I'm not very much a computer guy, so I can't really do that. But they had some sort of algorithm to design these different scaffolds, essentially mutating all the different bases while maintaining that secondary structure. How I designed my novel ones is that there's a software called NUPAC. And so some really smart computer people kind of made a really dumb, user-friendly version of copying and pasting a nucleotide sequence, figuring out its binding affinities, their 3D structure, copying and pasting that structure into a platform, and then it can just generate all these different variants. And so I just copied and pasted that and designed it myself via that program, NUPAC. Nice one, but with the obviously machine learning having more and more prominence, it'll probably be a really cool way to design even more guides and less troubleshooting behind it, because 90% of the guides are tested and aren't functional. So if you're able to take some of these functional guides, feed it back into the model, and kind of better, more rational designing of the future ones, save a lot of time.
Excellent. Thank you. Please join me in thanking Kyler. Very nice talk. Okay. You can leave it here. Yeah. Okay. Thank you. And our next speaker is Professor Mark Howarth. He's the Sheild Professor of Pharmacology at the University of Cambridge, Department of Pharmacology as well. Waiting for the slides. Yeah. Okay, here we are. Mark, thank you for being here.
Okay, thank you very much. It's really a great pleasure to have the chance to speak here today. And I just want to say at the start, a lot of this work has actually been carried out in my lab previously in Oxford, now in Cambridge, by people who've been trained at Imperial. And so everyone who's been involved in creating the excellent sort of teaching environment and generating the wonderful can-do attitude in molecular bioscience and synthetic biology, I want to really thank you at the start. So I'm going to talk about some of our lab's work on developing new kinds of protein interactions and exploring how that can empower basic research, develop potentially therapeutic tools, all the way through to the clinic and thinking about big challenges in infectious disease and global health. And so we are often looking for inspiration, and we've seen earlier in the talks today wonderful inspiration looking in nature and in the biosphere, but we can also get our inspiration from what happens in laboratories, synthetic organic chemists. And so my lab's been very inspired by click chemistry, which won the Nobel Prize a few years ago. And so that this is the work trying to get molecules to come together incredibly simply and robustly. Obviously there've been many sort of synthetic reactions before, but the real focus here is on simplicity, robustness, so a high modularity, so you can take two reactive groups. This is sort of the prototype. You have an alkyne and azide, and you mix them together, they react. But the nice thing about a sort of click chemistry reaction is you can have your alkyne attached to all sorts of different things, your azide attached to all sorts of different things, and the reaction will still work. And that's not the case for lots of chemical processes. So they also want very high yield. They want it to happen under all sorts of conditions, so you don't need to work in a glove box and have it completely dry, and then you can apply it in living systems. And also this is really important, starting from things that are easy to get hold of. And I'll talk about that with regard to biology. So what I want to introduce is this concept of click biology, where we'd like to have a few good reactions that can join biomolecules with some of these really advantageous characteristics. And if you think about click chemistry, the essence for which turned into bioorthogonal click chemistry, having selectivity away from all the natural chemistry in living systems, is they put in these exotic functional groups, which you almost never find. And so what I'm looking for click biology is, can we get robust reactions, but using the groups that every cell of every organism has got? So simple things, amines, carboxylic acids, hydroxyls, thiols. Can we lock things together in an efficient and selective way? And so these are the things that we want to achieve. We want to make it from regular biological building blocks, so actually not use our natural amino acids. Can we do this with the regular things that the cells have got? Have broad fusion tolerance, get really high yield, so things are very efficient. We want it to happen fast, because biology is moving, incredible speed. You blink your eye in a fraction of a second. There's a lot of biological events that have gone into that. We want a level of control that we don't have to wait hours for things to happen. We can exert rapid control. Condition tolerance. So biology is complicated. We've got organisms that survive in ice. We've got ones that survive in hot springs. We've got everyone focuses on pH 7. Well, you look in a eukaryotic cell, you've got the lysosome pH 4. You look in your digestive system, you've got the stomach pH 1 to 2. You've got in your pancreatic secretions, it's up at pH 10. So we want things that work under a range of situations. We've got oxidizing environments, we've got reducing environments. So it's nice to have reactions that are highly tolerant. And then we don't just want it working in our one favorite organism, we want to easily be modularizing it. So for people working on bacteria, for people working in mammalian systems, for people working in plants, we want a system which will be generally applicable. So that was sort of the challenge. Where do we start? So in this case, we start by looking to nature. We look at this bacterium, Streptococcus pyogenes. So this is something that probably almost certainly everyone in the room has been infected with this bacterium. Just causes a sore throat, strep throat most of the time. Occasionally causes more dangerous conditions. But it faces a challenge. So it lives in the back of your throat and your nose, and it needs to establish these biofilms, and it needs to avoid getting swept away as people sneeze and cough and the mucus is flowing. So it's got some clever anchoring mechanisms. And so it's got these adhesion molecules which can grab on. This protein called FBAB, it's got some very interesting chemistry. It forms a thioester at the N-terminus, which is a sort of reactive warhead. But along the chain, it's got some other domains. And this is the one that we homed in on as being particularly exciting, because it does this kind of protein chemistry that is really unusual. Normally you've got these side chains, lysine and aspartic acid. You find them on the surface of the proteins, they would be charged, and you just get a little bit of electrostatic interaction. But in this very specific environment that this has evolved in this protein domain, they will just spontaneously react. So it's a bit like GFP. The protein folds and the reaction happens. And it's kind of paradoxical, because normally if you want to form an amide bond, you've got to put some energy in. You activate it with ATP. But here you get amide bond formation. You lose water even in an aqueous environment. What's happening, so it's in the hydrophobic core of the protein. There's a glutamic acid opposite, so it's a kind of triad which helps facilitate the reaction and shuffle the hydrogen, the protons, and facilitate amide bond formation. So what Bijan, Jacob, and Emily in my lab set out to do was to turn this lock into a glue. And so all these proteins are named SPY from S. pyogenes. So we thought, how do we harness this chemistry? The simplest thing we can do is just split this protein into parts. So we take out one bit, which is the SpyTag, which is a peptide, and then we have the rest of the protein, which is the SpyCatcher. So the SpyTag, it's a short peptide, it's got this aspartic acid. The rest of the protein is the SpyCatcher, it's got the lysine. And so the vision was, you fit these two together, everything is perfectly aligned, and hopefully you can get this reaction to happen. And so the SpyTag will fit in the groove of the SpyCatcher, and that seemed like the simplest way to advance the chemistry. So sort of echoing back to the click biology criteria that I was talking about, what Bijan in my lab could establish is that we mix these two together and we get very high yield, so we can get up to 99% conversion. There's almost no side products to happen because we're starting with things that are pretty unreactive. It's just an amine carboxylic acid, what side reaction could happen. But also, we've got this robustness to conditions, so we can do this under all sorts of different pH, temperature, no particular requirement for anions or cations in contrast to most enzymes. And also, in this sort of test tube situation, quite fast, but this is when we put proteins together at 10 micromolar, so what would be typical after you purified them. Thinking about the challenge in biology, most of the things you're interested in are not at 10 micromolar. A typical protein expressed in a cell, it might be a low nanomolar range. So how does the reaction happen then? Well, if you start with the parent system, the original SpyTag-SpyCatcher, this has got a second order rate constant of 2 × 10⁻³ And so if both partners are at low concentrations, 40 minutes in and you've only got a little bit of coupling. And as I said, you want to follow, cells are doing things, well, that's not fast enough. So Anthony Keeble in my lab set about using directed evolution on both the tag and the catcher, combined with some computational stabilization and facilitating nice complementary interactions. And we got a second generation, the 002 pair, which is about 10 times faster. We got a third generation, SpyTag-003, SpyCatcher 003. And so this is now a lot faster. This is actually close to the diffusion limit for molecules of this size. So at that point, we thought, okay, well, that's doing pretty well. And so the consequence now is we've got both partners at low concentrations, and just in 10 minutes, we can get the majority of this to couple. So we can get this sort of reaction at a speed that fits in with most cellular expression levels and most cellular events. And so we can apply this approach, and we can generalize it, and we can look elsewhere in nature. Where else do you find this chemistry? So it's not just in Strep pyogenes, it's also in Strep pneumoniae, another gram-positive bacterium. And so we can take a domain of this, slightly different chemistry, so it's a lysine asparagine. So in this case, we have ammonia as a product, but we can apply it in the similar way. We can take out one peptide, and we have a SnoopTag-SnoopCatcher pair, or we can take out another peptide. The thing you notice about dog tag, it's actually a hairpin, it's not a linear strand. So that's very useful for putting it into tight turns in proteins. We can also direct it for peptide-peptide ligation. That's the Snoop ligase system. So we have these orthogonal pairs. They're not quite as fast. Dog-catcher is about as fast as the original SpyTag-SpyCatcher. So with this general approach, we started doing this. Other labs started employing the strategy, and there's now this big set of similar pairs. GSK decided they wanted their own pair that they engineered. So it seems like a generally applicable way to get irreversible interactions to peptides. So we've got a nice module. We can stick things together irreversibly through an amide bond efficiently. What can we do with this? It took us a while to come up with some killer applications, but we've been using it to stabilize enzymes. So we put SpyTag at one end, SpyCatcher at the other. You lock the termini, which are often the floppiest bits of enzymes. And those enzymes, which initially you heat them, or you give them harsh conditions, they unfold. When you cool them, they tend not to refold. They tend not to aggregate and not regain activity. We've shown for many enzymes, which we call spy rings, that in this case, having cyclized them, they can regain full activity after those harsh conditions. It really helps their refolding. We can interface with nanotechnology, with all sorts of cool platforms, gold nanoparticles, magnetic beads, bubbles. Great work done in Imperial, using this to anchor to graphene for cryo-EM. Also connecting to gene therapy vectors. I'm going to come on to other nanoparticles later. Thinking about mechanical biology, so this is the isopeptide bond that you form is stable to heat and high temperature, but is also stable to force. So for people understanding how force changes molecular behavior, you need something rigid to anchor onto, and SpyTag-SpyCatcher has often been that key link. And then in the biomaterial space, again, you've got substantial forces that you want to resist, but you want to do it in a programmable way. So this is nice work from Francis Arnold, Tim Liu, taking genetically encoding molecules with multiple SpyCatchers, another one with multiple SpyTags. Nothing happens to them. They're nice and soluble as is, but you mix them. And if you want to, in seconds, you can get them to gel, but it's completely programmable. So you can put in cleavage sites, you can put in interaction sites to integrins, you can put in growth factors, and then because it's specific, it's very cell-friendly to do this assembly. Thinking about sort of moving towards applications relevant to the immune system, which is a big focus in our lab, in terms of antibody technology, massively powerful for studying fundamental biology. A lot of us are operating at the level of 2 or 3 antibodies to study our favorite pathway. But the way the world is moving, we need to head to a situation where we've got at least 1 antibody to everything in the proteome of your favorite organism, and then, you know, one person's favorite organism may be E. coli, another may be a plant or a Drosophila or a naked mole rat. So actually the world is going to need tens of thousands of effective antibodies, and when you have those antibodies, you're going to want to use them in all sorts of different assays. So you might do flow cytometry, you might be doing ELISAs and you need it linked to an enzyme, you might want to inject in an organism and you need a certain circulation time or a certain reporter. So we also want very efficient functionalization of those molecules. And if we're going to do that genetically, say 50,000 antibodies, 10 fusions, 500,000 constructs to purify and characterize, that really doesn't scale very well. So much better if we can use a modular approach to link on the effector domains. So this is nice work initially developed by Jim Collins applying SpyTag, and I'm going to tell you how we've taken this forwards. So having a binder with the SpyTag, you make it once and then you can link it to all sorts of modules linked to SpyCatcher, fluorescent proteins, enzymes, toxins, and so on. So the way that we've taken this forward is not just linking a binder to an effector, but linking a binder to another binder, because a lot of biology depends upon bringing molecules together, and that's done effectively by bispecific antibodies. And these have had great success in the clinic. You think of BiTEs that are targeting the immune system to kill cancer. But I think there's an awful lot to discover about how molecules communicate and find new synergy. And extraordinary subtleties in how cells sense information. So there are many receptors where, as you pull them together, according to the changes in distance or the changes in angle, you get completely different things happening downstream in terms of how the cytosolic molecules sense proximity, sense orientation, and lead to different effects. So people have been very interested in this approach, but they're trying to work out, okay, how do we start to build these molecules in high throughput? And what has reached the clinic is using controlled exchange, breaking disulfide bonds, reforming disulfide bonds. And that's been quite powerful, but it really requires optimization every time you do it. So I'm going to tell you about the work of Claudia Driscoll, a graduate student in the lab, and she wanted to accelerate this process. And so this is what we call the SpyMask approach. And so the vision is that what we're going to do is we're going to get many, many proteins linked to SpyTag, so both antibodies, nanobodies, affibodies, even natural ligands like growth factors or signaling peptides. And then what we're going to do is we're going to put them into a double SpyCatcher, a double catcher. So we've got a SpyCatcher, a spacer, a SpyCatcher. Of course, if you've got 2 ones and you just add those, you're going to get a statistical mixture. We don't want that to happen. We want to get a precise system. So this is what Claudia set up. So we've got our first SpyCatcher, we've got the linker, and then the second SpyCatcher's got a mask on. So the mask is just a dummy SpyTag that has a mutation at the aspartic acid, so it can't react, and it sits in the groove, and that SpyCatcher is 99% caged from reaction. And then we've got a TEV site on a little loop. So what we do is we add the first ligand, it goes in site 1, and then we add our TEV protease, the little dummy peptide falls off, and then we can precisely get the second ligand in site 2. And Claudia could get that at 95% purity just in a one-pot reaction. So to give you an illustration of the kind of assemblies we can make, if we look at HER2, so this is a tyrosine kinase, overexpressed on many cancers. It's a target of a successful breast cancer therapy, which is Herceptin. So this is the Fab from Herceptin. It's binding right here. But HER2 is a very nicely studied system. There's binders identified to all parts of the surface. So there's different Fabs. Here's a nanobody. Here's an affibody. And so what Claudia did was just express each of these modules with a SpyTag, use the SpyMask system to plug them together in 96-well format, And then you add them to cells that have got HER2, a cancer cell line that depends on HER2 for its survival, and you see what happens to those cells. So very simple readout. And what you see is everything happens. It's very complicated. So red means the cells start dying. Blue means the opposite. The cells actually get stimulated to proliferate even more. And so I think this sort of illustrates the complexity of the different orientation, how you're recruiting these molecules, but also the need to explore at high throughput to enable us to find more synergy and really optimize the effects that we can have on the cells we want to control. And so the second part of this platform is thinking the limitation of IgG. So IgG, fantastic therapeutics, but if you want to achieve precision in how you grab onto molecules, well, you've got your FC, and then you've got fabs, and you've got flexible spacers. So these fabs are waving around, and they're not really sending the molecules exactly, their targets exactly where you want them. So what Claudia could do is, by making a set of different double catchers, we can actually tune where those fabs extend, and therefore where the molecules that they're linking to are oriented. So this represents the orientation of the SpyTag and therefore how the ligand will extend. So we've got these, this is sort of an initial set. We definitely want to extend this. But you've got a lot of complexity. You've got 3 dimensions of distance, and then for each of these, you've got a rotation. And so what you can see here is, again, blue means proliferating. So if we link trastuzumab to an Affibody, we make this bispecific. In this orientation, cells start proliferating. We change the orientation, and cells start dying. So we've got really a very large binder space that this approach can help us to explore and optimize. Okay, so moving further and thinking about immunology, I want to now start thinking about the stimulating protective immune responses and vaccines. My initial background was in immunology, then I went into chemical biology and nanotechnology. And from that perspective, I think vaccines fit very nicely because your nano orientation really matters to how the immune system considers a target. So if you take something completely foreign and you inject it into an animal or a human, then the majority of the time you get little or no immune response. And that's not by chance. That makes sense because your body is exposed to lots of things in your food, you breathe in, and you don't want to make immune responses to all of them because that would give you pretty bad allergies, autoimmune diseases. But if you can take something foreign and give it a nano-sized orientation, so sort of 20 to 100 nanometers, if you have high-level multivalency on the surface of that, then the situation is very different, and your immune system takes it very seriously, and you're likely to get a strong immune response. So why does that happen? With that size, you head to the lymph node, which is the key place to kick off immune responses, is someone with a swollen lymph node. When that particle interacts with a B cell, which is going to be key to make the antibodies, which are usually the most important things for successful vaccines, you get high-level clustering. You may get activation of innate immune responses. And so actually, from a nanotech point of view, making a vaccine is somewhat straightforward. So of course, there are many different ways that you can build vaccines, and people are now very familiar with these different viral vectors or mRNA platforms. In my lab, we've been very focused on virus-like particles because we think they have a very high safety margin. They can be very potent at eliciting antibodies, and they're also very scalable, that you can make billions of doses of these very, very cheaply for a few pennies a dose, and they can be very stable. So essentially, we have a protein-based virus-like particle platform, no nucleic acid in, it's not going to evolve. We have an antigen, we want antibodies to it, and if we can decorate it like this, then we probably may have a good vaccine. So that's the concept, very simple. And then the challenge has been that many of these antigens are not very nice proteins. They're marginally stable. They've got complicated modifications. And so you do your genetic fusion, and things turn very stressful and keep failing. And so your particle may not assemble. Your particle may not have the native conformation. You may not be able to get it in high enough yield. And it's really a very, very painstaking, slow process, and it's not really what we want in engineering biology. Then we talk to the chemists, and they come up with some cool coupling technologies, but then you tell them, how cheap it's got to be, and everyone uses a very simple technology, which is cross-linking agents that would link an amine to a thiol. And so you get somewhat heterogeneous particles, and you can publish lots and lots of papers on these, but these are not getting approved. These are not good enough, really, to fool the immune response, to give you the potent antibodies in a reproducible way. So this is where Linda and Carl's a former Imperial student, came in, and thinking about what we call plug-and-display vaccination, taking this modular strategy, building the virus-like particle once with SpyCatcher, and took some engineering to do this. This is starting from a phage protein. We later moved on adapting some of Neil King's and David Baker's computationally designed nanocages, and then further developing, engineering them ourselves for high resilience. But essentially, we can make very large amounts, very cheaply, of a SpyCatcher virus-like particle. And then, generically, if you've got your targets from different pathogens, as long as you can make that antigen with a SpyTag, then it's just a case of mixing it in. You will get your SpyTag-SpyCatcher isopeptide bond formation. You can get it at high yield under all sorts of conditions and have something ready to kick off an immune response. And so we started doing this on malaria, collaborating with Simon Draper and Shimi Biswas in Oxford Jenner Institute. And since then, other labs have really picked up on this and shown that this modular strategy can work for them in a big range of different viruses, can work in different bacterial proteins. You can even get it to work on self-targets. So because virus-like particles are so stimulatory, you can break tolerance. You can take self-proteins, put them on, and drive immune responses. Why do you want to do this? Well, in cancer, you may want to have binders to checkpoint molecules, these overexpressed antigens, or even personalized medicine, things that are unique to an individual's cancer. You may be able to drive immune responses, both antibody and T-cell based. And so it's great that that's been picked up on, but we want to go beyond publishing papers. And so now this has entered the clinic. So Simon Draper has started a phase 1/2 trial. He's working on a blood stage antigen for malaria vaccination. Cytomegalovirus is a very common disease, but there's no good vaccine. It's the most common cause of deafness and blindness in babies. And so a company I was involved in founding, Biotech, this is now in phase 1, so we're really keen to see how that will advance. And then AdaptVac have taken a SARS-CoV-2 into phase 3. So this tag-catch is slightly different tag-catcher technology, but it's already been tested in 4,000 people. So that's sort of the known viruses. I want to talk a little bit about this guy, a horseshoe bat, which has got lots of viruses that you can sequence, and they're not yet a problem for humans, but you can see that there are not many mutations from being a problem, and we need to think about how we can actually do even better than the community did for the first COVID vaccine generation. So, why do I say there is going to be a next pandemic? Well, I don't know if there'll be a pandemic, but there will be new viruses emerging into humans. We've had SARS in this coronavirus family. We've had SARS-2. We've had MERS carried by camels. And you look in the bat sequences, also interesting work done in UK bats. You can also find viruses that have some relevance here. Pangolins, this Pang-17, have a virus in this family. So we've got this challenge. There's a lot of viruses out there. with potential for transmission. So we started collaborating with Pamela Bjorkman and Alex Cohen at Caltech. And the question Alex asked was, okay, if people have had COVID infection, we've got these viruses are a bit related, do those antibodies actually stretch out and give you protection across these other viruses? So the y-axis here is how much antibodies you've got. So if you've had COVID, you've got antibodies to this receptor binding domain, the key bit that grabs onto the cell receptor. So everyone's got antibodies to that. But you look to a very close bat virus, so there's the RATG13, and you've got almost no antibodies to that, and very little, but it's pretty much close to background for these related viruses. So people probably are not very well protected should another virus emerge within this family. So what Alex thought was, well, can we build a vaccine which would give that broad protection? So he took the SpyCatcher MI3. MI3 is our preferred nanoparticle. It's full of holes. And so this is what people have done before, just link on the SARS-2 RBD with a SpyTag. But what Alex did was just make many of these viral RBDs with the SpyTag. You don't need to get hold of the virus, you just need some sequence. And all you've got to do is mix them, and you get this, what we call a mosaic virus-like particle, and you can plug in 4 or 8 as you prefer. And what's the idea? So the principle is we're trying to sort of do some sort of logic gating at the surface of these B cells. So if we hit the B cells with a uniform virus-like particle, they only need to stick on one bit of the surface. The B cell receptors get cross-linked, which is the key to activate them, and that B cell will be stimulated to divide, will make lots of antibodies. But those antibodies may just see one of the targets. In this case, the different colors represent different RBDs. Only if the B cell receptor sees features that are common between the different colors are you going to get strong stimulation, strong activation. So we're providing some kind of evolutionary pressure for the B cells that have that cross-reactivity. So to give you just one little flavor of the data that we see from this, what we do here is we make a pseudovirus, so a virus that's not dangerous, but we can monitor its cell entry ability, bearing this bat RBD. This virus is called SHC014. We immunize mice and see how good those antibodies are at protecting from infection. So if we take the SARS-2 RBD alone, after immunizing mice, that's no good. That's not above background. We take that protein, put it on a virus-like particle, well, that definitely helps. We get some protection. If we are not sure what virus is coming, and we're a bit worried about this one, so we have it in a mixture of 4, then with that mixture, we still get excellent antibodies to the SHC014. But I think this is the most interesting one. This doesn't have the SH014, but it's got some relatives. So this is thinking about a situation where we don't know what viruses are emerging, but we know some of the family tree. We know some of the relatives, so potentially we can start to think about building these vaccine platforms before a new danger emerges. And with this mixture, we actually get very good, pretty substantial neutralizing antibodies from that sort of preemptive vaccine. So this approach has got funded by this Coalition for Epidemic Preparedness Innovations, and this is moving very soon into clinical trials. So we're very excited that this is a pretty unusual thing to take into the clinic. It's unusually complicated, so we've actually got 9 different components to get the best protection. We've got our SpyCatcher MI3 cage, and then we've got these 8 different viral proteins. And so, very interested to see how that will develop, but thinking, can we do better for the long term? So, I want to tell you about the work of Rory Hills, also a graduate student in the lab. And so he's been making these tandem versions where we can actually take multiple RBDs and actually use genetic fusion. This is quite a tolerant domain, and just giving you a flavor of it. So if we look at antibodies to the SARS-1, the original SARS, which was not in our assembly, comparing homotypic, so just putting one RBD on, or these mixtures, so this is pretty much the one that's going into clinical testing, we can actually, with this quartet nanocage, we're calling it, actually get even better antibodies elicited. So overall, I've tried to introduce this concept of click biology, having very simple, fast, selective reactions to help synthetic biology move fast. People do things that they can't do with classical methods. I've told you about SpyTag-SpyCatcher. Of course, there's other reactions that are fantastic, split inteins, halotag, snaptag that you may have used that I think meet some of these criteria. And I think it's important to reflect on what are the key characteristics for us to move forwards as a community in advancing these technologies, making them powerful for people. But with the SpyTag, we've tried to make a tool that would be very useful to people in all sorts of applications. I focused on irreversible locking, but biological systems, often dynamic assemblies are useful, and so we've also built pairs for SpyTag-SpyCatcher that are reversible and have tuned affinity, and that's been employed in the cytosol. We've shown that we can make a switchable SpyCatcher, and you can use that for purifying your protein, so you don't need to use a histag. You can purify off SpyTag. We've used light control. We've built a toolbox of assemblies to give you controlled oligomerization. And then we've also working on this networking to using SpyMask to very rapidly explore binder space. And in terms of medical applications, we've exploited this and made this broadly available for people to build these tools to existing major challenges and maybe newly emerging ones. And it also sort of suggests a different approach to vaccine design. We have all these tools from genomics now, very rapid surveying of sequence space, and I think some of this work shows that the real possibility of starting to build these vaccine platforms to certain families and really being pre-armed. So I'll stop there. Very grateful to all the members of my lab. As I said, many of whom were trained in Imperial for their key contributions. Really grateful to Alan Townsend, Simon Draper, Shimi Biswas for their help on the vaccine development, and Alex and Pamela Bjorkman. And a shout out for SynBio UK. Hope to see many of you there. And thank you very much for your attention.
Thank you, Mark. Great talk. Questions?
Hello, interesting talk, but one thing I want to ask, because it's never seems to do with the vaccines, is there was quite a bit of hostility to vaccines during COVID, and one of the reasons was needle phobia. Are they actually going to develop a vaccine on these lines which can actually be orally administered like a sugar lump like they do for polio? Because that might overcome some vaccine hesitancy.
Yes. I mean, that's a big question. It's been an area of massive interest. People have been thinking about it for a long time, trying all sorts of things. The challenge has been that, as I talked about before, the immune system is not responding to everything. And so if you're breathing things in, you're eating them, a lot of the focus is actually to achieve tolerance to those. And so when people have made vaccines that are not delivered through injection, they've either not stimulated strong enough and not got a reliable, 95, 99% protection, or they've had to use different platforms, which, and this is really what's gone through, is attenuated viruses, which are have other challenges. So it's something we'd all like, but there's some sort of serious immunological challenges to that.
Can I just make a quick response to that?
Yeah.
If, say, the immune response is only 50% rather than 90% with the oral administration, surely that's better to develop it for people who don't like needles than it is simply not to only have the jab and then people refuse it and they have zero protection if they don't have it.
Yeah, I mean, there's important sort of issue there of whether you have a sort of choice and like a buffet system, and when you're getting a vaccination, or the government decides, okay, this is the most effective system, can we employ it? But yeah, it'd be great to chat later and get further perspective.
Thank you. It's very interesting that it's very versatile. You have shown us a lot of examples. So I'm sorry if I missed it. I want to ask about the conjugation part. So how complex is the conjugation of this catcher and tag to each of the components that you're going to couple later on?
Yeah, so it's all through genetic fusion, and because the reaction's happening through the side chains, then you can put it at the N-terminus or at the C-terminus, either part, and that's perfectly fine. We've been exploring in some cases you can also insert it at internal sites in proteins if that's most effective. So we've tried to build in, that simplicity of simple genetic fusion to arm the parts, and then you just mix them together.
It's being done in the DNA level?
At the DNA level, yeah,
Okay.
exactly.
So is there any consideration on there might be possibility of change of structure, right? Because I'm not sure I could see that the catcher, for example, it's the size is not really big, but it's also rather big. Is there any consideration on that possibility of the change of structure?
So, I mean, most of the time, we've now tested it on in collaboration on a large fraction of the yeast proteome, and so I think we got to like 85% through those. So I think it's not working 100% of the time, but by and large, most proteins are pretty tolerant to that fusion. Occasionally, we just change the size of the linker or swap it to the other terminus, but generally high robustness we're seeing.
Thank you.
Thank you.
Do you see any trade-off between the performance of the vaccines as you start to generalize them more? Like, do you feel as they get more general, do they become less good at
right.
performing?
I mean, we're very worried about that, but it's not really what we saw, actually. Having the mixture in, you get almost as much antibodies to the target. The trade-off comes a little bit when you focus in at the molecular level, and you start to think about where on your particular target you want the most of your antibodies. And what you find is the bits that are most protective, which is exactly where the receptor's binding, are the most variable, that has been the most evolutionary pressure. And then the bits that are most conserved often are not fully protective. So that's something that we're currently working on, trying to optimize that trade-off, but it's a great point.
Okay, thanks.
Hi, great talk. Can you use these structures, these mosaic structures, in the context of creating a multivalent vaccine? For instance, dengue, where you have the 4 serotypes causing the disease. So how do you control the expression and avoid the AD effect?
Okay, well, I don't think I put dengue on my list. So dengue is really hard target. It's also a bit different because it has a protein shell, and which is naturally multimerizing, so it doesn't lend itself that easily to putting on another self-assembly. And as you say, you've got this ADE, so I think for things that are self-assembling, I think this is maybe not the best approach. We're doing some other work, as I was just mentioning in response to the previous question about focusing immune responses that are separate to SpyTag molecular assembly. So I think that kind of molecular engineering is the next step for dengue.
Hi, Mark.
Yeah.
I think, if I'm not right, original version of CRISPR also comes from S. pyogenes, is that right?
That's true.
And I think there is some evidence or concern that a lot of humans already have antibodies against some of the components against CRISPR. Have you ever seen any issues with that, people having antibodies against SpyTag-SpyCatcher?
So our focus has been to use it not as a therapeutic, but as a vaccine. If you're working with a vaccine and you're building on something, you want that thing not to be human, to avoid any danger of autoimmunity. So for pushing the vaccine applications, then I think that's perfectly fine. And we've also done experiments to show that if you have those antibodies or you made more of them, it has no effect on how much antibody you get to your new target. But you raise a great point because with this molecular assembly, we're very interested in therapeutic options through these bispecifics. And so what we're exploring now is using some of these computational tools to actually turn this bacterial protein and try and camouflage it and hide it from the immune system, both at the surface level and, because it could get processed by T cells, remove these good targets. So that's the next big advance. Thanks.
Thank you, Mark. Really enjoyable talk.
Thank you.
You mentioned just at the end developing reversible reactions. So my question is around, do they come from the original SpyTag-SpyCatcher system, and under what conditions is the reaction reversible?
So just to clarify, we stop the reaction happening and we get a non-covalent interaction, but then we drive reversal. And so the way we've done this is we focused on making a pH switchable one. And so what we did was we put in a load of histidines very close to the SpyTag on the SpyCatcher, and there's 2 positive charges there. And so at neutral pH, everything fits and it's all fine and hydrogen bonding. And then you go to pH 5 and suddenly you got all these positive charges and we get very nice release. So that's our preferred way in the SPY switch to elute. But what we found at that point is we also had built-in temperature switchability. So at 4 degrees at pH 7, it interacts quite nicely. We keep at pH 7, but we go to 37, and it dissociates very nicely as well. So that's a sort of orthogonal way that can be convenient.
Thank you.
Thanks. Very interesting. And I'm wondering if there is a version of the system which is essentially can self-ligate some short peptides. Is there a way to ligate short peptide, kind of to make it circular?
Cyclize.
Yeah.
I guess we could do that. You may well be familiar with the split intine system that's fantastic at doing that, that you can do very short peptides and get these wonderful molecules you can genetically encode, and Alitavasoles has massively advanced that. So that's not been our focus, but I just want to highlight that. Yeah, other very interesting chemistry around.
Thanks.
Okay, thank you. If you have no further questions, no, then please join me in thanking Mark for an excellent talk and discussion. Okay, and with this, please join us for lunch outside, and we'll continue with talks and the sessions just before 2.
yeah, just try and keep them as close to your mouth as possible. But there have been, people have been having them down here and they're still been thinking that you need to be there the whole time. There is a clicker, sorry. Yeah.
Dracarys. Okay, everyone, welcome back to this second session of this symposium of the Institute for Molecular Science and Engineering. My name is João Cabral. I'm a professor of soft matter engineering here in the Department of Chemical Engineering, and I'm also one of the associate directors of IMSE, and so it's my pleasure to chair this session, and invite our first speaker, who is Cinzia Clem. I just practiced earlier on. And to start with, we'll have two contributed talks. These will be relatively short talks. And then we'll have an invited talk, which is slightly longer at the end. I will ask you, if you don't mind, to refrain from asking questions during the presentations, of course, but keep them to the end. And whenever you ask the presentations, please introduce yourselves as you start, just so that we can put everything in context. There will also be people who are joining us online, and so they will be submitting questions too, so please keep them coming, and I will be managing those questions and sort of place them orally on your behalf. So if I could invite Cinzia to take the stage and ask her to start on telling us about understanding and optimizing phenotypic heterogeneity in S. cerevisiae. Over to you, Cinzia. Thank you.
Thank you so much for letting me present my work today. 1 second. Yes, so my presentation will be about understanding and optimizing phenotypic heterogeneity in Saccharomyces cerevisiae. And I am a postdoc in the Rodrigo Ledesma Amaro lab. And our lab is focused generally on driving a more sustainable bioeconomy using microbial organisms and engineering microbes to generate sustainable solutions that are not only better and more sustainable, but also cheaper, or at least competitive in pricing with traditional methods of synthesis. And we're led by Rodrigo Ledesma Amaro, and our group is constantly growing in the past years. And the RLA lab has been working to produce all these different compounds in recent years, and also worked on engineering yeast to utilize cheaper feedstocks. And we have multiple different pillars in the RLA lab to optimize, I don't know why this happens, to optimize our research and to drive our research. And one of them is to engineer synthetic biology tools to optimize engineering. Then we work on engineering bioproduction to generate yeast that can produce specific molecules that we're interested in. We work on microbial communities to understand how communities work and how communities can be used for microbial bioproduction, engineering, and also, for example, to use these for division of labor. And specifically in my project, I'm working on phenotypic heterogeneity and basically understanding why cells are behaving the way they do. And now, it already jumped ahead to my next slide. What is phenotypic heterogeneity of clonal microbes? And basically, we describe phenotypic heterogeneity as when a cell has a certain phenotype at a specific frequency, and what we want to be this phenotype like is I don't know why keepers aren't happening. I'm just letting it jump ahead if it wants to. Sorry, can this be changed? I think it was, maybe it can be. Sorry. It maybe is like, is it selected on auto jump or, okay. I don't know why this happens, I'm very sorry. This is fine. So basically, what we want is a distribution of our phenotype that is just unimodal, and basically we have most cells having the same phenotype, producing in the same amounts when we look at bioproduction. The problem is what we usually see, thank you, hopefully this works better, is that we have either macroheterogeneity, which can be described as, oh, it still happens, sorry. So it's either macroheterogeneity, where the phenotype, where phenotypes can be present in two states, in a, basically show as bimodal distributions, or we can have microheterogeneity where a trait or phenotype is present in a quite big variation within cells. Maybe it's easier if you jump. And phenotypic heterogeneity of clonal microbes is basically often leading to lower bioproduction yields as when we scale up these systems from like a smaller scale, or if we let the cells grow for a longer time and induce them to stress. Usually these small populations are actually outgrowing the high-producing populations because they have less resources that they use for bioproduction, and they can use more resources for growth in general. So now, I don't want to talk too much about genetic heterogeneity. This is not what we are interested in. I mean, in general, genetic heterogeneity is derived by differences in the genome, but we'd like to look at non-genetic heterogeneity, which is driven by epigenetic events, microenvironment, and microenvironmental changes, such as differences in nutrient availability within a bioreactor, differences in gene expression, or cellular noise. And then a few examples for production heterogeneity are, for example, Saccharomyces cerevisiae cell size or protein abundance. And we also saw from our own research in the lab that there's different bioproducts that have different levels of heterogeneity, such as beta-carotene production in S. cerevisiae or lipid production in Yerevia lipolytica, as well as indigo-iodine production in Yerevia lipolytica. As you can see, the beta-carotene, which is autofluorescent, is very variable in different cells, as well as lipid bodies or indigo-iodine, which is the blue dye in the Yerevia lipolytica. So the heterogeneity team has been growing since 2020, consistently with multiple people working on different projects, such as Davina, who worked on promoter heterogeneity, Kian Shabestri, who worked on nitrogen source quality. Both projects were a little bit more fundamental, and now we kind of move into a more applied field, looking at using heterogeneity sensors to study bioproduction heterogeneity, looking at proteomic heterogeneity and cell signaling that can optimize bioproduction, and studying like examples for bioproduction heterogeneity, such as lipid production in Yerevia lipolytica. And so the first topic I'd like to talk a little bit about in my project is heterogeneity on the proteome level. And so for now, we mostly study heterogeneity in single cells on a more computational level, on a more overall level, using single-cell RNA sequencing. And the problem of single-cell RNA sequencing is that we obviously have to isolate single cells, do the reverse transcription, amplification, and sequencing. And problems with this is that a lot of post-translational regulations aren't detected at all. The samples are super sensitive, and the data is very noisy, and the coverage is quite low. So there's a lot of problems that we can't actually detect what we really want to see. And so what I've done is to use a GFP tag collection of yeast strains, which consists of more than 4,000 strains, and each of which has one specific protein tag with GFP. And we made this collection prototrophic to then induce different phenotypes by moving these strains from rich media to starvation media and incubating them for a few hours. And then we've analyzed these strains using flow cytometry and microscopy to see whether the protein distribution has changed using flow cytometry, or if the proteins redistribute within the cells. And with this, we want to then identify heterogeneity markers, different metabolic profiles, and do target identification for potential targets that we can optimize in our bioproduction processes. And so this is basically just the overview of the data that we've gotten out from these screens. And what we see is that a lot of different pathways are actually heterogeneous when we expose them to stress and different environments. And for example, the steroid biosynthesis and lipid metabolism pathway are also heterogeneous, and these pathways are quite important for a lot of the biosynthesis pathways that we are interested in. So this is quite interesting and promising for us to look at. And interestingly, many of these targets are also regulated by post-translational modification, especially by signaling pathways and proteins involved in stress signaling, for example. And so in my second part of my project, I'm basically using beta-carotene as a case study to identify heterogeneity and maybe find solutions to actually improve the production heterogeneity we observe in beta-carotene producing strains. And I chose beta-carotene because it's quite a simple pathway to integrate into yeast, and also because it's autofluorescent, so we can nicely track it with the flow cytometer, and it's quite nice within the cells producing these nice little foci that are actually accumulated in lipid bodies. And so yeah, I've done then flow cytometry with this. Sorry for the jumping, it is really annoying. And with the autofluorescence, I could see that after roughly 48 hours, when basically the glucose is depleted in the culture, the cells turn to be heterogeneous. And I also saw, when I now plot the beta-carotene autofluorescence compared to size, that this is size related. So basically cells that are smaller have usually much, much less beta-carotene levels compared to bigger cells with a bigger cell size. So we now argue that probably there's a specific mechanism that changes the metabolism of the cell or arrests the cell in a different way after the active growth stage that then induces this different phenotype of beta-carotene accumulation within the cell. And so because we saw the size difference in the phenotype, I looked at different genes that are involved in cell signaling and growth, and I deleted these genes to see if I can change the heterogeneity phenotype and if I can see improved beta-carotene accumulation. And actually, when I deleted SNF1, which is the major glucose signaling kinase within cells, I could see not only an increase in beta-carotene abundance, but also less heterogeneity, or at least my large population was suddenly much larger. So this is quite promising, and it's quite interesting that SNF1, which is sensing that the cell doesn't have any carbon, is also the culprit of causing this heterogeneity. And now, obviously, we don't want to delete just SNF1 and use this for bioproduction because it's a very important regulator that has a lot of different targets, but we can now uncouple the signaling pathway of SNF1 that regulates beta-carotene bioproduction from SNF1 signaling. And that's basically now going to be the next steps of my project. And yeah, with this, I'd like to conclude that bioproduction heterogeneity is widespread, and starvation causes, in many cases, also heterogeneity, or triggers, is one of the triggers for heterogeneity. Cellular adaptation is often mediated by different cell cycle signaling proteins, and we can take our data to optimize, to engineer optimized strains that are sort of insensitive to these signaling pathways. And with this, I'd like to thank everyone in my lab and our collaborators, and thank you for listening. And sorry for the slide chaos.
Sorry. Thank you, Cinzia, and I'm sorry for the hiccups with the tech. Our apologies. Do we have time for a couple of questions? Chris.
Thank you for the presentation. So I saw that you have heterogeneity with respect to size and to beta-carotene levels. Were those beta-carotene levels absolute, or are they normalized to biomass? And if they're not, do you think that it's strictly as a matter of cell size? So essentially, if you have product per biomass, is there less heterogeneity along that metric rather than just looking at beta-carotene?
So there's a tiny, we did actually look at that, and it's still, I mean, the problem of the data with the flow cytometer or with the FACS is that there's so much data that if we take two populations, there's always going to be some significance. It is not super linear, the distribution, so you still see that there's like a little bit, a non-linearity if you plot cell size to beta-carotene autofluorescence. But in general, and when I actually do sort these cells into a low and high producer population, I see smaller cells, and these smaller cells also have usually just one of the foci, so they have much less lipid bodies. And if I then do microscopy for quantification, I can see a difference between cell size and biomass and beta-carotene levels, but it's, the cell size phenotype is definitely the most strong that leads to this difference in beta-carotene levels.
A question at the back there. Thank you.
Hi, thank you for the talk. I saw that you use the single-cell transcriptomic data to show your proteome level change. I wonder whether you think that's accurate because, and also whether you can use single-cell proteomics to do that, whether that's more appropriate?
So I think we didn't really want to, I mean, obviously we believe that our system has like some advantages to single-cell transcriptomics because, single-cell transcriptomics is quite hard to do with microorganisms because they don't have a lot of transcripts. And we don't get as much coverage as we want to. And I get that obviously our system is very different. We look at GFP levels of tagged proteins, but I think it's still quite valuable because we can track so much more proteins. We have it in live cells, so we can really look at it whilst it's happening and not just after like the purification and after sorting of single cells. And we actually saw that in a lot of the proteins, the data is very consistent. So we did compare it to transcriptomic data sets, and we do have the ones that are heterogeneous in the data set that were published with transcriptomics. They are very identical to what we see with the proteomics in some cases. And I think, I think it's just sort of a very nice adding to the existing data.
Yes, because we are running a little bit late, and so that we try to get back on time, could we please thank Cinzia again for an amazing talk? Thank you. Sorry, I think it was our fault. If you could leave the mic here, Cinzia. And could I invite Jose Jimenez Zarco, who's a reader in synthetic biology in the Department of Life Sciences, who will present to us on engineering bacteria to eat plastic.
I've got my own. I think, it's working fine. Can everyone hear me well? Yeah, perfect. Thank you. Thanks a lot for the invitation. It's a great pleasure to be here, and good afternoon to everybody. I'm going to switch gears slightly. We just introduced a reader in life sciences, and my group has a strong focus on synthetic biology or engineering biology applied to environmental problems. So what I want to present today is some of the work that we've done related to trying to use microorganisms or enzymes to break down plastics, and this was in the context of a European project. And the first thing that I want to do is to introduce the two main responsibles for all this work, who are Alice Banks and Umar Abdul Muttalib. Both of them left my lab, and actually Alice was meant to give this presentation today, but she left 2 weeks ago. She took a principal scientist position in a biotech company, which is very exciting. So I'm going to do my best to give the same presentation as she would, although I'm pretty sure it's not going to be as good as she would have done it. Anyway, moving on, I don't think I need to convince anyone about the issue that we have with plastics. We have a problem with plastics. We are making lots of them. And what you have in here is just a picture, it's a projection of how much plastic we expected to make by 2050 if we keep business as usual. Just to give a sense of perspective, more than half of the plastics that have ever been produced were made between the year 2000 and 2020. Okay? The problem with plastics is, we make a lot, but also it takes for them ages to degrade in the environment. So they are not biodegradable at all. And actually, this has created a great environmental concern. And about 5 years ago, almost 6 now, there was an issue of National Geographic trying to raise awareness about this problem that sadly came wrapped up in a few layers of polyethylene film, but that's a completely different story. But since then, many people have tried to make an effort to find solutions for the plastic problem. Now, The other thing that I want to mention is that plastics are very completely different animals. So when we talk about plastics, it's not just one type of polymer. Actually, we have many types of polymers that we use on a daily basis, and they're all different. By different, I mean their chemistry is different. And this conditions greatly whether they can degrade at all or not. So we have different types. If you see in the news that there's a new enzyme that breaks down plastic more efficiently, I'm almost certain that it's going to have to do with this one here that is called polyethylene terephthalate, PET, which is a polyester. So the reason why we have enzymes, so this one is very hard to degrade, and my talk is actually going to focus on it. But the reason why it's easier to degrade than the others is just because of the presence of these ester bonds. So in nature uses esterases a lot for very different functions, and some of them happen to have the ability to break down polyesters, like this one here. Now, for other types of plastics, like polyolefins that have carbon-to-carbon bonds, the chemistry is so much more difficult that it's really, really difficult to find just a single enzyme that might be able to break down this carbon-to-carbon bond. Okay? So most of what you see in the news probably relates to esterases. Sometimes we also see news about amidases. Again, breaking down polyamides is easier compared to other types of bonds. So here I'm just presenting one solution for polyesters, okay? But you may have heard of many others, and actually for some of the molecules we have really good recycling methods. So this can be mechanical, they can be chemical, some of them are biological. And this is one example of them. There's a company called Carbio in France who have developed the technology to break down plastics like PET enzymatically, and this works at an industrial scale. And again, you may have seen some of this in the news. So they use this enzymatic process to break down the PET, get the monomers, and then reconstitute them and make fresh polymer. So that's great, and it works with plastics that we can actually collect. And for PET in some countries, especially in European countries, we have high collection rates. But I also want to mention that there are lots of plastics that sadly we are unable to collect. Some of them are also polyesters. So we use a lot of PET, for example, for making textiles. These textiles, through the process of wearing down, washing machines, etc., eventually end up in wastewaters. From the wastewater, they end up being forming part of microplastics that end up in the environment, just maybe through disposal, through sludge, that can be used as a fertilizer or polluting different types of waterways. So ideally, what we'd like to do is to develop a technology that is able to tackle not only the plastics that we can collect, but also those that are really, really difficult to collect. And that's why we thought it might be really interesting to come up with some sort of organism that might be able to produce these enzymes that are needed for breaking down plastics in situ. And why not, might be able to use the plastic as a feedstock for growth. So effectively, that they could eat plastic. So to our knowledge, there is only one organism that is able to do such a thing. It's called Ideonella sakaiensis, and it was isolated in Japan in 2016. And what it does is it makes two enzymes that are called Paez and metase that are secreted and break down the polymer that releases the two monomers that then can be assimilated by the organism. Now, the problem is that this organism does so with very low efficiency. And actually, the organism is really picky and it's difficult to work with in the lab, not to mention that it's almost impossible to do any sort of molecular engineering with it. That is, at the end of the day, what we want to do. We want to improve this process. And therefore, we set on the task of trying to engineer microorganisms that ideally should be able to do the very same thing. And this is what I'm going to try to explain next in not so simple steps. So what we want to do is to generate a microbe that can make these enzymes that break down the plastic. The plastic generates the two monomers, and then they are assimilated by the microorganism as food, and then can grow on them. Now, there are a few hurdles that we need to overcome. So the first one is we need to be able to have organisms that can use either or both of these two substrates for growth. The second issue is we need to engineer efficient secretion of these enzymes outside of the cells. And for those of you that work with gram-negatives, you may be aware that it's really difficult because they are not great secretors of pretty much anything. The third step is we need to find an enzyme that is active enough in the concentrations that they are being produced by the organism and in the conditions in which the organism has to grow in order to break down the plastic. And finally, and this is crucial, we need to be able to find the plastic that can actually be degraded. And not all of them is possible to break them down with enzymes. Therefore, we need to identify pre-treatments that are good enough to support microbial growth. So the first step was kind of easy. We just went on campus and we isolated microbes from different environments using traditional microbiological enrichment. So basically, we take a chunk of soil, we incubate in the presence of whatever molecule we want the microbes to use as the soil carbon source, and we wait. And if we wait for long enough, and if we do that several times, we end up with a few of them. We isolated the whole collection. All of them are able to degrade either one or the other of the components of PET. Some of them can actually take both. And you have some examples of them growing on terephthalate or ethylene glycol as soil carbon sources. We like a lot these ones here that are Pseudomonas, because these are organisms that we know how to manipulate very well in the lab. And again, you have an example of how that works here. So ideally, we want to not only be able to take the plastic as carbon, but if we could transform that plastic into something else of value for biotechnology, it would be great. So what we do is we take the violacin biosynthetic pathway, and violacin is a molecule that gives this distinct purple color to the colonies. It has antimicrobial or antitumoral properties. For us, the important thing really is that it's purple, and then we know if things are working or not very easily. And then if we plug those genes into some of these organisms, and we have 2 of the environmental isolates here, what you can see is that the terephthalate, represented in yellow, can be consumed by some of them, and in exchange, we get some of the purple production, which is great news. Okay, so microbes is sorted. Next step is to try to engineer the secretion. And this was a bit of a challenge, so it took us a while to find out what is the right way of doing it, but eventually, we found the secretion leader that can be cloned just in front of each of these enzymes. And then if we grow colonies of the microbes, and we grow them in the presence of a mock substrate for PET, it's another polyester, but it's slightly soluble, it's called polycaprolactone. If an enzyme is active and it's being produced, then we should see clearance halos around the colonies that you have represented here. So our favorite one is this called PHL7, because it's with the one that we can get the highest activity in these particular conditions. So far so good. Now we can secrete the enzymes. Not all the enzymes are equally active against the substrate, and actually some of them are not active at all. And that's what brought us to change the assay completely, and then instead of using polycaprolactone, we switched to something that is slightly easier to degrade for all these enzymes. That is called phenylbutyrate. What matters is when the enzymes break down this molecule, they release something that is yellow, and we can monitor very easily. And what we decided to do was a side-by-side comparison of all the enzymes being produced just by E. coli and taken from the culture supernatant, and no purification involved or anything like that, to see under growth conditions which one is going to be the most effective for us. And again, we did all those assays for different temperatures and for different types of enzymes. Some of them come from thermophilic organisms, some of them have been described in the literature, some of them have been designed by artificial intelligence, like this phosphatase that you have in here that again made the news not that long ago. And in the results, once again, PHL7 is the best. And now that we know which one is the best, is the one that we can carry forward for the next step. The next step, super important, that I mentioned at the beginning, is the pretreatment. As I said, plastics are very different, but even the way in which we use these plastics to feed the reaction is important as well. So we tried a few methods, and we ended up with 4 of them to try to determine whether they're good substrates for microbial degradation or not. So we tested films that are completely amorphous. One of the problems with plastics is that if they're very crystalline, they don't break at all. So this is amorphous PET that we can buy from a company. We cryomill this PET, and this is equivalent to what is used in the industrial process. We also sift this cryomill PET to try to get even smaller fractions that should have a higher surface area exposed for the enzymes, and this will be representative for microplastics in the environment. And we took some bottles and trays from the supermarket, so this is post-consumer PET, and what we did was we dissolved them with an organic solvent that I'm not allowed to say what it is yet, because it's been patented by some collaborators. But effectively what happens is, as a result of this dissolving into the organic solvent, we end up with a very rubbery, very thin PET formulation. Now, if we once again repeat the enzymatic breakdown of all of them, we get the results that you can see in here. The colors of the bars represent the different plastics used in the process. You have the different enzymes, including the controls here in the upper part of the plot. And the height of the bars represents the amount of terephthalate that can be released from the plastics. And in the results, there is a very clear difference between the solvent-treated PET, the red bar, compared to the others. Actually, after 3 weeks of incubation, we can barely detect anything coming out of the standard PET plastics. But for the pretreated one, and especially working again with PHL7, we see a release of terephthalate that is in the millimolar range, and that's good enough for the microbiologist in the audience to try to grow microorganisms using PET as the sole carbon source. So once that we know, then we try to put everything together. We combine the best pretreatment, the best enzyme, and the best microorganism. And then we did growth assays using PET as the sole carbon source, and you have the results on the screen right now. The red line represents the culture of the microbe when there is no PET. The blue line represents the culture when there is PET. Here we record how efficiently the microbes grow. This is just for the microbiologist, again, it's a way of monitoring growth. And what you can see is that there is just a tiny difference. So if you've done this experiment, you know that this increase is not impressive. So doing everything the best that we can, the increase in biomass that we can get is about 2.5, and that's about it. So Alice wasn't very happy with these results and repeated the experiment about 1,000 times. And I'm showing the results on the screen as well. So this is the increment in OD comparing with and without plastic for the empty plasmid control. And this is when we have the proper enzyme. So there is a modest increase, but this modest increase is real. So doing everything the best that we can, this is all that we can get. So just to conclude, this work demonstrates by combining pretreatments, systematic investigation of what's the best enzyme, and using the best microorganism that we could find in nature, well, at least out of the ones that we screened, we can get a modest PET trophy. So this is what people like to call using PET as the sole carbon source. So as a bottom line, if you are worried about plastic eating bacteria released into the environment and eating all the plastic, I'm sorry to tell you that's not going to happen anytime soon, based on the results that we've got so far. But if there is something that nature does very well is optimizing things by using evolution. So who knows in the future, but as of today, I wouldn't be too worried. So I want to finish just by thanking all the people in the lab that you have on the screen at the moment, our sponsors, and of course you for your attention. Thank you.
Thank you, Jose, for an awesome talk. Do we have time maybe for one question so that we gain some time back? Question at the back there.
Hi, very nice talk. I was just wondering if you had thought about performing some mutagenesis studies on the PHL7 enzyme, which is that the wild type or is that a mutant?
Yeah, that's the wild type. The only thing that we've done to it is to engineer the secretion leader, but we haven't done anything else to it ourselves. Our collaborators that discovered it in Germany have done some evolution, some directed evolution studies, but we haven't changed the sequence that we originally got. But yeah, there is potential to make it even better, hopefully.
Second question is, quite often with plastic degrading enzymes, they are quite insoluble. Did you do some sort of modification, like solubility tag or something, to try and solubilize them if you're just using the overexpressed cell lysate for these degradation studies?
We haven't done any modifications to them. To my knowledge, they're purified as they are overproduced in E. coli, but that's not exactly what we wanted to do. Actually, our plasmids the expression levels are moderate because we want them to be secreted outside, not so much to purify from the inside. So we are using just as they come out of the cells, effectively in the supernatants, whatever concentrations we get.
Okay, thank you.
Can I ask everyone to thank Jose once more? I'm sorry to cut you a little bit short. And can I invite Claudia Contini, who's a lecturer in life sciences, in biotechnology and engineering department. And she, significantly to us, she's also a L'Oreal Women in Science awardee from 2022. And she'll tell us, she's our invited speaker for this session, and will tell us about synthetic cells from soft matter to cell-like behaviors. Over to you, Claudia.
Thanks. Is it working? Yeah. Hi everyone, I'm Claudia, and I'm a new PI in the life science department. And I just started my group next year in November, so I'm a new PI. I'm very happy to start this new adventure with my amazing research group, which I will acknowledge at the end of the presentation. And so what we are doing in my group is something different from what you heard before in the previous presentation. As my title says, I'm a lecturer in engineering biology, but what we are doing is actually to create cell mimics. So we take inspiration from nature, from cell, and we create our synthetic biomimic system. And in this presentation, I will give you just an overview of the research vision, why we want to mimic something that already exists, and also some successful projects that are the foundation of my research group, and also a very short overview of all the projects that we currently are carrying out. And if you like anything that I'm going to say, please feel free to ask any question, to reach me out during the event, but also to send me an email or contact me via email or Twitter. Okay, so we mimic nature, but why mimicking nature? So this is exactly the same question that I was asking my father that was painting what was exactly already there in front of his eyes. And I was not understanding why I was spending so many days, months to actually try to mimic, reproduce that something that he could get with just a camera or was just there in front of us. We just needed to open a window. And what he told me, his answer was very simple. He just told me, sit down here and look, it's beautiful. And what actually I think that he meant is that only when you mimic nature, only when you try to reproduce it, you can actually understand its complexity. And because you can understand its all different details and all the different components that make nature as we know it. And so nowadays, scientists are basically like artists. We take inspiration from nature, and scientists can mimic useful things. So we can create synthetic hearts, synthetic hands, synthetic animals. And all of this is not only to challenge science, but also to create a new biotechnological tool that can help, can be useful for a real-world application. But this is not a new trend in science. Scientists have been always taking inspiration from nature. For example, Leonardo da Vinci, 500 years ago, was observing how birds fly and was translating that biological observation into engineering tool that were going to allow the man to fly for the first time. And over the year, science evolved. So we passed from the ornithopter to the Antonov, that is the world's largest aircraft. And in doing so, in this evolution of science, actually help us new way of locomotion and allow us those new ways before were completely unthinkable. And actually, in this evolution, we learn how to perform better than nature. In fact, in a normal flight, London to Sardinia, we are already flying in 3 times faster than the bird with the greatest airspeed velocity. And all of this started just mimicking nature. So I've been told that in South Kensington, I will not have enough space to fit an Antonov. So we are not working in macro scale, but we are going to work in more micro and nano scale. So if we look at this video, this is actually an example of high sophisticated behavior that we can find in nature. What you are seeing is a droplet of human blood. The spherical static object are our red blood cells. The black dot moving around are bacteria. And the large cell, sorry, I'm going to replay it. The large cell that you see there, it's our immunocell. So our immunocell doesn't have any eyes, but what it does is moving to a specific direction, and what it does is translating the chemical signal, specific chemical signal that come from the bacteria into mechanical motion. And this is a process that is called chemotaxis, and it's part of our immune system, but also take part in tissue growth and so on. And so imagine now to create a synthetic version of that system. So a synthetic cell capable of a directional autonomous motion in our human blood. Well, it will help us to do different things. First of all, to understand the challenges of the motion in biology, and also for an engineeristic point of view, which are the minimal set of compounds that require, that lead to the motion of a nano or micro object in physiological environments. And also we can translate that knowledge to create new biotechnological tools that will help us to have smart active drug delivery, so a system that specifically is attracted by a specific chemical signal and move directionally to that specific target. So in that case, we get rid of all side effects. And then we can use the system also for biosensing, because we have a performance that is activated by a selective biomarker. And also we can create a smart material, because if we think engineering collective behaviors of motile system, then we can control the assembly and disassembly. So how to do that? Well, in my group, we use what is called bottom-up synthetic biology approach. So we create our synthetic cell, or probably it's more correct calling them a minimum model system or cell-like system. We create them from scratch, so we take molecular tools, we control their chemistry, physical chemistry, we self-assemble them together, and finally we have our minimum model system that can mimic the cell structure, function, and properties of cells. And in my lab specifically, we use any molecular tools. So we are not restricted to biology. We are not trying to reproduce the origin of life, but we are going to use any molecular tool available to mimic cellular behaviors. So in this way, we use biological parts, but at the same time, we can use, for example, synthetic polymers or inorganic nanoparticles. And after we create our system, well, we play around with the soft matter engineering, and we can create smart materials, as I said before. We can also use them for bioremediation, because the system is capable to translate a signal from the environment. We can use them, again, for drug delivery and sensing, and also will allow us to understand which is the minimal set of compounds that lead to sophisticated behavior. And this is because I mean, for everyone that works with cells knows very well that cells are very difficult to manipulate and to understand, because even if they are considered a unit of life, they are actually very complex. And in a minimal system setup, instead, you have a system that is 100% controllable. You know exactly what is in there, and you can study which molecule, which signal pathways participate to one specific event or cell performance. So when we want to mimic the cell, if you look at the scale, one of the first steps to mimic a cell is to achieve a degree of compartmentalization. So we need to protect the internal environment from the external environment. There are different ways to achieve that. What my group uses are mainly two molecular tools, the lipids and polymers that are amphiphilic, so they have an hydrophilic and hydrophobic part. We control the chemistry, how they interact one to another, and the supramolecular interaction, and at the end we have our minimal model system, or vesicle, liposome, or polymers. And when you engineer the soft matter, then you can start controlling interesting parameters. For example, we can control the morphologies, can be spherical, shape, or multi-compartment. We can have different architectures, like layer in a layer, or vesicle in a vesicle. And then we can control an interesting parameter, which is the surface topology, so the surface of organization of our system. So this is exactly how I started my professional career. I was working in bottom-up synthetic biology before knowing that I was working in bottom-up synthetic biology. So I was making my polymer from scratch, and in order to make my polymeric vesicles. And what I learned from here is that by controlling the chemistry, the exact chemistry of your molecules, so you can actually control the final properties of your vesicle. For example, you can control the thickness, the permeability, or stimuli responsiveness. And in this study, for example, where I participated with collaborating with Teoni group in the material department, one of our students was synthesizing 3-block copolymer, where the hydrophobic block was of different length, and at the end we obtain membranes with different thickness. And this is actually an interesting parameter, because if we modulate the thickness, we can modulate the membrane elasticity, but at the same time the membrane permeability. And this is another example where I was using the same chemistry, so it was the same copolymer, to make different morphologies. And this was possible only playing around with the physical chemistry. In this case, the amphiphilic copolymer had two parts, one hydrophilic and hydrophobic. An hydrophobic part has a tertiary amino group that can be protonated and deprotonated. So if it's protonated, the chain is fully hydrophilic, there is no self-assembly. If it's deprotonated, then you have the self-assembly and the formation of self-assembled structures. And this amino group has a pKa, they change with temperature in a way that decreases when the temperature increases. So if we solubilize this polymer at a pH that is lower the pKa, and then we slowly increase the temperature of the solution, we can actually see the formation of different self-assembled structure. So we have the nucleation from micelle to spheres, and then to what we call the hygenous structure, but you can call them also pretzel as you like. And this morphological change is actually is possible to characterize using different techniques, NMR, calorimetry, and so on. Sorry. And another parameter we can control, as we said before, is the topology. How we do that is mixing two amphiphile, they can be two copolymer, two lipids, or lipids and copolymers together. They have a different molecular weight, they have different chemical, physical properties. They mix together, and over time we can see the formation of domains on the vesicle surface. And this is a very interesting parameter because we can not only create, use a domain selective functionalization strategy, but also the final structure will incorporate the properties of two membranes. So we have two different permeability and two different stimuli responsiveness. And all of this is fantastic, but if we think a real-world application, this will be already enough for a drug delivery system. In this study, for example, we tested different shapes of assemblies from sphere and tubes, and what we saw, for example, that when the cell uptake happens, the tubes causes enhanced metabolic stress in cell compared with spheres. And so once we engineer our minimal model system, then we can use it to understand biological properties, for example, the membrane biophysics, or how the membrane interact with external objects like proteins or inorganic nanoparticles. So in this study, we are investigating the interaction of different proteins that are different by molecular weight, but also hydrophilic, hydrophobic profile, and how they interact with polymeric membranes of different curvature, so micelles and spheres. And what we saw, we are seeing is basically the proteins interact more with micelles, and this is an interesting effect because a protein corona, it's one of the challenges that need to be overcome in order to create a drug delivery system. And the reason why they interact more with micelle instead of spheres is because at the point of contact, the protein with the spheres, the grafting density is very high, so the polymer creates a spherical encumbrance for the protein to interact. Instead, for the micelles, there is more space of interaction, and the protein can enter in contact with the core of the structure. Another interesting part is understanding how inorganic nanoparticles interact with membranes. So Jose was telling about a microplastic nightmare. We have also a nanoparticle nightmare because nanoparticles are basically everywhere, are in our food, in our cosmetics, and also in our future with the nanomedicines. And even if they are terribly useful, we don't know their long-term effect in our body. If we think about the multiple ways they can enter in our body, where there are many. So one of the first toxicological steps to investigate is how actually inorganic particle can interact with membrane, and if can actually penetrate the membrane. And when a nanoparticle is put in contact with a membrane, we can have three different scenarios, or only one that is a sequence of the three. So we can have no interaction. In that case, we have a Brownian collision nanoparticle with membrane. We can have nanoparticle absorption in the membrane, and that is characterized by penetration depth. Or we can have the full nanoparticle uptake. And this is when the energy of absorption is much larger than the energy cost associated with the bending of the membrane. And the last part is actually something that happens in real cells without any active transport. It's just a part of the membrane biophysics. So when you have enough absorption, enough interaction, then there is a nanoparticle full uptake. And this interaction investigated with liposomes. So the minimal model system was a vesicle made of lipids, and I made it interacting with gold nanoparticle of different sizes. And what we discovered is that there were two critical radius. One 20 nanometer, where before that the particles were aggregating on a membrane surface or being uptaken with a tubulation effect. And then another critical radius around 50 nanometer, where afterwards the nanoparticles were just bouncing on the membrane. And this because the different interactions were leading to increase of the surface tension and were interacting much less. But the amazing part of this study is that all this collective aggregation, in particular the uptake by tubulation, was only theorized in literature. There were no evidence of this. And thanks to the use of the minimum model system, we were able actually to visualize this collective uptake. And another interesting collaboration that I want just to mention is led to a paper in Advanced Material where basically there were biologists in Slovenia understanding the interaction of inorganic particles with cell lungs. And this is very important if we think that workers in the working site constantly breathe in those nanoparticles. So they wanted to check if there was any interaction between these materials and cell lungs. And they asked me, can you help us? Because when working with biological systems, they were not sure what exactly was interacting with what, and if the uptake was driven by protein or just was a simple membrane biophysics. So it was a very interesting interaction between a bottom-up synthetic biology and biology. So after we create our minimum model system, we can sophisticate a little bit more to achieve life-like behaviors. And to be honest with you, one of the behaviors that I really like and I'm really obsessed with is motility. So as in the video, creating this synthetic chemotactic system that can move to target direction. And one successful example of chemotactic vesicle has been realized with what we call asymmetric vesicle. So we achieve a degree of asymmetry in our nanovesicle by mixing two different copolymers. So we observe the formation of domains on the vesicle surface. And at the end, the vesicle were formed by two domains, with the smaller one being the more permeable one. And the reason why we want asymmetry is because in the nano and micro scale, we are in the low Reynolds number, so the viscous forces are dominating, which means that the water creates a resistance to the motion of the object. So you need to have an asymmetric system in order to apply a propulsion mechanism that push the nano and microsystem in solution. Plus, we need an external stimuli, like a chemical gradient that sets the directionality. So here we achieve our asymmetry. And then in this asymmetric vesicle has been installed a protein and gene. So are basically two proteins that collaborate together in a cascade reaction. And what is happening here is that as soon as the proteins produce a product molecule, they are ejected for the more permeable domains. And this asymmetric distribution of molecules around the vesicle set the condition of self-diffusion phoresis and motion of the vesicle. And I'm going to show you this motion macroscopically. So in this experiment, the Petri dish has been filled with a sample. And then at the center of the Petri dish has been injected the substrate. And what we can observe over time is the collection of the vesicle by chemotaxis at the center of the Petri dish. I'm just showing this data, but just to be realistic with science, to actually make this video took me 4 years of optimization in the lab. Okay, this another motile example has been done in CES lab, where I had the great luck to co-supervise Xiaobin, a great PhD student. And what we were doing is using, using an emulsion made by two solutions. One solution is PEG, a polymer, and the other solution is made by dextran. When you mix this solution, you form an emulsion, which droplets are not stable, so you need to stabilize them. And we stabilize them adding liposomes to the formulation. And these liposomes sit exactly at the surface of the droplet. And the reason why is because they are functionalized with PEG, that is the same polymer that is in the continuous phase. And there is actually an optimal concentration of PEG that allow for the vesicle to sit at the interface. And then when to this solution, with this emulsion, we add the dextran, we just see the vesicle merging with the main droplet. And if we add the PEG, we have just a dilution effect. But if we add water to the system, then we start to see the droplets moving. And if we pay attention about this motion, we can see that it can be divided in two steps. In the first part, there is actually the formation of the asymmetry, which I told you is very important if we want to create a motion in microscale or nanoscale. And then only after there is an optimal asymmetry, there is actually the motion of the droplets. In this case, away of the gradient. So what is happening here? As soon as we add the water to the system, we create a polymer gradient on the continuous phase. And this causes the migration of the liposomes by Marangoni flow. And this, and as soon as there is the migration of the liposome, then we have the creation of another gradient of interfacial tension, and they finally push the droplet in solution. Another interesting effect is when two droplets meet. So when they actually collide with each other, they don't have anymore the liposome protection because they are already moving, and then there is the merging of them. But during the merging, there is another liposomes rearrangement in the surface that stop the motion and only take place again when we have again the asymmetry forming in the droplet. And then I'm going to just mention a project that is about creating a polymer-based artificial cell, and we are trying to engineer dynamic behaviors with polymeric vesicles, and this is a shameless promotion of the poster of my PhD student in the room next, so feel free to ask him any questions. And so we are using a D-block copolymer that is temperature sensitive in a way that has a phase and transition from lamellar to hexagonal. And with this polymer, we can create beautiful, stable vesicles that keep their size even after months, are very, very stable and very easy to work with. And, but as soon as we increase the temperature of the solution, then we have this transition in the vesicle. And what is happening is that on the top left, you can see that we don't see anything in the microscope, but then as soon as we increase the temperature, we can start to see the nucleation. And then when we cool down, we have actually the formation of micro objects. And what is happening in here is that when you apply the temperature, you have the phase transition that causes the membrane deformation. So the vesicle in solution and nanovesicle in solution enter in contact, stick one to another. When there is the cool down, there is the final assembling one to another, and then we can form this multi-layer sponge-like microsystems. And those systems are very dynamic. So if we play around with temperature, we can actually control some dynamic behavior. One of them is actually the fusion in microscale. So even those multilamellar droplets, if we stay just below the transition temperature, it's enough to cause this membrane deformation, as you can see from the surface. And it's not playing, yeah. So they enter in contact, there is the fission between them, and then as soon as you release the temperature, there is the swelling back and fusion, and then a sharing of content. So one of the young part will be actually to use this system to activate a reaction through fusion, so two compartments that have different contents can actually fuse and activate a reaction. The good is that we can run them also in cycle, so you can run multiple fusion events. If we go instead above the transition temperature, we have another dynamic effect, that is the contractility of the system. So a system that is capable to shrink and then swell back in the original size. And again, an amazing part of this is that when shrinks can release whatever is inside, and when swell back can trap object in the external environment. And an example of this is a an experiment I'm going to show. I'm sorry if you're working in bacteria, you are bacteria sensitive, but I have to warn you that bacteria have been treated very bad in this experiment. So I was working with bacteria and decided to mix those particles with bacteria and applying in a heating and cooling cycle. And what you can see is that after doing this cycle, all the bacteria are trapped in those microsystems, so effectively cleaning all the solution from the bacteria. And I think that this is a very powerful behavior that can be used forward to not only to functionalize our system in a very easy way, but also to detect if there is, for example, selective trapping of the external solution. And again, a shameless promotion. There are different things that we are doing with the system now, so please feel free to visit the poster. Just to give you a very brief overview of what is happening in the lab, there is Shuan. It is synthesizing a polymeric capsule with pores, a different shape in nano and micro scale, and we are going to functionalize them with polymers brush in order to open and close our pores. There is David in collaboration with Ravinash in infectious disease, and that is 3D printing layers of droplets, and we want to create a sort of a signal pathways between layers. Nina finished her master and now is a PhD, but during her master I was co-supervising with Yuval Elani, a PI in chemical engineering, and we created hydrogel capable of magnetotoxic and deliver a bacteria cargo. There is Karina that is incorporating inorganic particle with our synthetic cell. Then Max and Sandra. Max is a PhD. Sandra is a master in collaboration with James in chemistry, James Hindley. So we are making hybrid vesicles. Max will move that project towards a motility project, while Sandra is focused more in domain selective functionalization. And then Joan is finishing his PhD very soon, and I co-supervise him together with Yuval Elani, and we are making biohybrids. So it's a bacteria that is dotated of a vesicle coating, and we use this coating for multiple purposes. One of them is to give magnetotaxis, but also to shield the bacteria for external components and so on. And then Amanda that is encapsulating bacteria into vesicles to understand if we can have, for example, morphological changes of vesicle with the bacterial activity, but also how bacteria respond to compartmentalized environment. And this is an ask for collaboration. So if you are interested in biohybrids, so conjugating biological or synthetic parts together, please let's have a chat. And I'm very interested in any way we can complex our system a little bit more in order to mimic more biological structure and functions. And so this is thank to the founder, thank to the amazing group that is with me in this adventure, and thank all of you for your attention.
Thank you for this awesome talk. I have a permission from Amparo to postpone a little bit the start of the next session, so we'll shrink the panel discussion. So we'll have time for maybe 2 questions, and then we'll have a 10-minute break and then come back. So any questions for Claudia?
Hi, thank you. I was wondering, with these polymer vesicles, are you able to put proteins in them? Can you have biological molecules interact with them and actually embed in them? What's kind of that kind of aspect?
Yeah, so with the polymer, this is the reason why I went also towards a polymer minimal system, is because I want to include a little bit more chemistry. They are more flexible in design, let's say that. And so, so proteins can be attached to polymers. embedded in a membrane, I guess that you need a protein membrane to do so, and is what Sandran is doing with a hybrid system. So we are incorporating protein membrane in actually the lipid part of the vesicle, but incorporating again the properties of the polymeric membrane in the same structure. But yeah, you can do multiple things. And we just started, so we are just playing around with the physical chemistry of the polymer to achieve those dynamic behaviors. But if a protein can be useful to sophisticate the system a bit more and achieve any other functionality, yeah, we can do that.
Thank you.
Any more urgent questions? If there are no urgent questions, can we thank Claudia, Cinzia, and Jose again for an awesome session this afternoon? And let's have a break. So I think we'll be back in 10 minutes, and coffee is outside, and we'll be calling people back in promptly.
a lot more because
Good afternoon, everyone. If you're ready, please take your seats and we'll make a start.
Do we have to flick a switch or
Okay, perfect. Good afternoon, everyone. Welcome back from the coffee break. My name is Cleo Kotorabi, and I am going to be chairing the panel discussion this afternoon, which is on challenges and opportunities in engineering biology and an outlook discussion on what the next 5 years hold for this field. As I said, I'm Cleo. I'm from the Department of Chemical Engineering, where I'm Professor of Biological Systems Engineering, with a primary interest of how to marry computational approaches with wet lab experimentation to get to the optimal solution faster. And together with me today, I have Professor Tom Ellis and Dr. Gogol Guha, and I'll hand over to Tom to introduce himself.
Hi everyone, yeah, I'm Tom Ellis, so I'm a professor in the Bioengineering department. I research synthetic biology and all things synthetic biology from an academic point of view.
Hi everyone, I'm Gogol. I'm the Chief Scientific Officer at Multis Biotechnology. If you don't know us, we work on cultivated meat and specifically creating growth media that can be used to enable advances in the cultivated meat area. And just not to undermine my credibility, but I don't have quite as much experience in this field as my fellow panelists, but I'm very excited to share some of my insights from maybe a more industrial perspective.
Perfect. So we have a few questions that we're going to go through and discuss as a panel, and then we'll open the floor to questions from the audience. So the first topic we'd like to explore is that of new technologies in the field, and specifically we would like to discuss which developments in the past 5 years you found the most exciting. Shall we start
Maybe I'll
with
go
Tom?
first.
Yes,
yeah.
please.
Yeah, 5 years, when you're as old as me, 5 years is like just like that, right? So I've got to be careful. If I say CRISPR, then everyone will be like, no. That's like 12 years ago or something now, right? I think obviously one of the things you can't ignore would be the use of AI and machine learning, right? And particularly we've seen in biology, in engineering biology, that has shown fantastic results in terms of being able to predict protein structure. And particularly in the last year or so, that initial kind of wave that came with AlphaFold has led to things more like CollabFold and other things in that wheel space, which really allowing people to start designing proteins. So no longer just about predicting the structure of proteins, but now thinking about designing from scratch using machine learning as an aid, proteins that bind specific things or that could catalyze a reaction that doesn't exist in nature or is suboptimal in nature just by trying to bind a specific intermediate in a pathway. I think that's very exciting, and especially if you kind of match that with the continual emergence of nanobodies and alphabodies, these sort of smaller versions of antibodies that are increasingly easy to make through biomanufacturing processes, then it's kind of exciting development there that we can sort of AI design small proteins that can be very good specific binders to do all sorts of reactions.
What do you think is missing there from AlphaFold? I have my own thoughts as somebody who's worked on glycosylation for a number of years.
Well, glycosylation. Yeah.
What do you think is missing there? Because, I mean, having seen some of the presentations, I guess a lot of the predictions are confirmed by proving the opposite. Almost like, okay, if there is, let's say, a prediction that a certain deletion will change the function, somebody will make that deletion and then will confirm that functionality is lost. So how do you think we can get to confirmation that the structure is actually correct?
Yeah, it's a good question. I mean, I recently was talking to some researchers who, particularly in this space, work on nanobodies and using machine learning in this. And you know, the current kind of like AlphaFold things are very good at trying to narrow down the area to investigate for specific binding of something. Like if you wanted to get like an antibody that was a great target for a receptor on a cancer cell, then it really reduces the space you have to test in for that binding. And what it completely doesn't reduce the space of, though, is if you do get something that's a very good specific binder to some protein, then actually to make use of it in the clinic, there's all these other things. It has to be manufacturable, it has to be easy to express, it has to be stable at all sorts of various conditions, so that it can be handled in the clinic and all those things. So I think we're missing that, right? AlphaFold and those related programs have been fed training data on structures. And so they're very good at structures. And the next sort of 5 years, 10 years will be like, can you put in the data sets on stability, thermotolerance, expressibility, glycosylation. Glycosylation's always the scary one.
Right, thank you. How about you, Gogol? What do you think has been the most important development of the past 5 years?
Yeah, so I think, like Tom said, AlphaFold and its impact really can't be overstated. I think that it's very hard to ignore the shift that, well, obviously machine learning is having in our lives at the moment. And I think the way it's impacting structural biology and the prediction of protein structure is just incredible. I think maybe to build a little bit on what you were just discussing about the enabling of this technology to actually lead to real breakthroughs in the way that we use these proteins that were either novel or predicted through AlphaFold. I think some additional enabling technologies that are being created right now are in actually looking at single proteins and looking at how they interact with specific ligands that may be predicted to be interacting with a certain binding site within a protein, like Tom was saying. And I think that these technologies have come a long way quite recently, where oftentimes the traditional approaches would require a heavy amount of modification to single proteins in order to be able to actually see what's happening to them when they're interacting with a different molecule. But the more you have to modify a protein, the further away it gets from the actual in silico prediction that you're making using a technology like AlphaFold. So things that require less labeling, like there's some recent technology that's been driven forward quite a bit, which is using light to trap single proteins. And if you know about optical tweezers, classically they're used for larger structures. But the ability to trap a single protein and look at its dynamics and how it interacts with ligands and other molecules, I think this is an incredibly powerful validation technique for something like AlphaFold, where, like you say, they train on data that's already been proven, and that's great. Lots of people put a lot of time into looking at these structures and making these connections. But now, in order to use it for something new, we need to be able to validate quickly and efficiently and build some trust in new results. So we're not, looking at these and saying, oh, it might work, it might not work. So I think in particular, technologies like this plasmonic optical tweezing, like I was mentioning there, or nanopore technologies that can look at single proteins and look at their dynamics, I think these can be very valuable to aid in exactly what you were talking about with AlphaFold.
And for somebody who is new to the field, are there any developments in high throughput production technologies for different protein structures or different variations of the same protein, for example, that have mutations or different translational modifications? Do we have that capability to investigate this in a single structure level?
Yeah, so it's being built, and I wouldn't say it's all the way there yet, but technology, at least on the nanopore side of things, from the company Oxford Nanopore, they've developed high throughput platforms to be able to essentially have a large number of parallelized membranes that you can use to create individual nanopores. And that's a nice platform for being able to look at different variants of a single protein, different mutations that you might have synthesized based on predictions from AlphaFold, in order to be able to actually run a relatively controlled experiment where all other conditions are the same, and then you are just changing that single thing and ideally not modifying the protein in any way. I think, at least from my knowledge of the optical trapping side of things, this is a little bit earlier on and less parallelized, but I think there's huge potential for that to be translated into something that a company takes hold of, or with enough effort from enough people or academics who grab hold of this to create a parallelized system, because it's something in principle that there should be no boundary when you're, the optics, if they're set up in a precise enough way, could be parallelized in a way that I think hasn't been achieved yet.
So assuming that we can really build on AlphaFold's capabilities and we can have the high throughput experimentation that will allow us to really validate the results, what do you think can be achieved in terms of translation within the next 5-year horizon, Tom?
Well, I certainly feel we will get therapeutic molecules that are binders coming out from de novo designed proteins. Obviously, if it's a therapeutic, it'll have to go through regulatory pathways, so it might be 5 years from now, it might be doing quite well in one of the phases of the clinical trials, but maybe not all the way through. I think, there's so much more to a protein than just its structure and its shape. So like I said, there will be the next set of things to look at. I think in the last 5 years or so, there's been a lot of increasing use of a method called deep mutational scanning in terms of trying to understand genes and their functions. That's where you use mutation either through synthesis, you make these libraries, and then you express all of these versions that have 1 or 2 mutations in various different positions in parallel, then do a massive assay to see which versions don't work. But you can actually do that in vivo now, well, within cells, through CRISPR-based approaches where you use CRISPR to target the gene and you put mutations all the way and you make a big library and then see which ones perform. Those methods are very good at pinpointing maybe on the structure of a protein, or if you think about a protein as a globe or whatever, all the other little places on the protein which are really important. So they often will reveal that something that AlphaFold could never reveal, like there's a patch somewhere on the side of this protein which is really important for the way it might dimerize or the way it might interact with other proteins or with antagonists or agonists. So I think if we can imagine that AlphaFold might be able to learn from the sort of, things like AlphaFold could be trained on those kind of data sets, and you could start to be getting much better understanding of drug target proteins and how you might be able to interact with them and modify those.
Do you see any obstacles in using AI now for translation? I mean, obviously, recombinant therapeutics have been on the market for decades, and it's widely acceptable that we use engineered microbes or engineered mammalian cells to make them. I guess because we tend to use them for patients that are already sick, there are no question marks, which is the complete opposite to what we've seen with vaccine hesitancy post-COVID, essentially. But now that we're moving into this era where we're heavily relying on machine learning, artificial intelligence to design molecules, or even hopefully in the near future to run processes, do you foresee any regulatory hurdles in that process?
Yes, I certainly can. So particularly in the UK, if we think about the regulatory sides of working with engineered cells, there are a lot of the regulation is based on introducing foreign DNA sequence that is not native, or an engineering, and it becomes more and more complex. And we've just in filling out our own safety forms in experimental labs within a university, we hit a bit of a brick wall sometimes when we're saying, okay, we're introducing a new gene, and then one of the first things on the list is what organism is this gene from? You say, okay, it's not from any organism. They say, okay, well, what is it sort of the closest relative of? And then there are some cases where, like, no, we are literally trying to make a protein that is built from scratch, where we're not taking and trying to mimic this. And so then the regulatory side sort of already starts hitting a roadblock at that point, let alone if you start to then try and take it to the next level where you start to manufacture it, or you might want to use it in the field or use it in the clinic. So I think there's some issues there that will need to be overcome, where there's going to need to be more of an understanding that it's not always about borrowing things from nature in the way that we're doing things in engineering biology anymore.
And Gogo, what is your take from an industrial point of view?
Yeah, I mean, I think Tom hit on quite a few of the things that we deal with on a regular basis. I think that consumer acceptance, I think drives a lot of this, at least in the food industry, where people shy away from things that are engineered in favor of things that are natural, even if things that are natural aren't always guaranteed to be safe or safer than things that are engineered. But it's very hard to convince somebody of that, because the things that are natural have been around for a long time, and the things that are engineered are completely new and unknown. And I think one of the things that we're seeing in the cultivated meat industry, and I think this applies quite a bit to any of the breakthroughs that AlphaFold is perhaps catalyzing at this moment, is that at the beginning, when a new technology like this comes out, there's a lot of excitement and a lot of potential, and everybody's like, oh, this could solve all these problems, and we don't have to make any compromises. We're going to create the perfect solution that everybody's going to accept, and nobody's going to have any problems with it. It's going to revolutionize this field, whatever field you're talking about. But I think once you get a little bit further forward of that and you start actually doing the applications, you start to realize, oh, it's very difficult to actually solve all the problems without introducing hurdles in terms of, oh, this thing has some regulatory issue because it's from, it's closest to the human genome sequence, and this is something we don't want because, we don't want to introduce this into a therapeutic that you're using with humans, or it's something that's completely unknown and we have no idea how it's going to interact. And I think what we're going to realize is how much effort has to be put in, how much has to be invested into the type of studies that will convince people, because there's really no shortcut to that aspect of it. I think if you have a technology that you think is great, you could prove it to yourself and feel very confident in it, but to gain mass consumer acceptance, you have to do the studies. You have to design safety and regulatory studies that you can create and publish that people will look at it and say, okay, I believe it now, where I didn't maybe believe it as much before. And I think that's perhaps the only way to really address some of these challenges.
Mm-hmm. And given that we don't really have marketed products on the supermarket shelves that come from engineered microbes in the form of alternative proteins or cultivated meat products, is there a regulatory framework that is reasonably well defined in Europe, other than to say no GMO?
Yeah, I think that it's challenging because every country, every group of governing bodies has their own frameworks, their own things that dictate how you should operate, and you're trying to find for us at least, as a mostly scientific company, you're trying to find the best approach that addresses the biggest portion of the market so that we can get in somewhere, rather than trying to make it work for everyone. So I think largely for us, that's been looking at the FDA, because they're, in a lot of ways leading the pack in terms of at least the way that they've been approached and the direction that the conversations are going are a little bit further forward than some of the European governing bodies. So this is what we're looking to, just as a single target, as like, okay, this is the best, most furthest advanced place that we can look for. And they have, because these conversations have been going on for some time with companies like Upside Food, there are, it's basically forcing the creation of actual standards that people can meet. Because, yeah, this is completely new for everybody. Nobody has anything in place. We have to create it. And I think the people that are pushing this forward, the cultivated meat companies, at least in this area, are making those happen by advancing those conversations.
I mean, regulatory, it's just a nightmare, right?
Well, it is. I think
it's different in every different country. I'd say, from what I've experienced and seen with people who've tried to get engineered microbes as biosensors against arsenic approved for EU regulations, I'd say it seems EU is mostly probably the hardest. The EU grew out of basically a cartel of European farmers trying to fix prices so they didn't compete against each other. So as long as it's in the interests of the traditional farmers of Europe, then it really gets a lot of support within the EU, and anything that is not within that sphere is difficult. And it doesn't even have to be engineered, right? So I heard a case recently, there's a lot of great potential use of filamentous fungi as meat replacement. But a lot of the same organisms for this filamentous fungi in their fruiting body form are pathogenic, but that never happens in the cultivation and then
sterilization of them to make these products. But because of that, it's basically going to be very hard to bring them to market because there's legislation in Europe that really strongly controls these organisms just because of one particular phase in their lifestyle, that they are related to pathogens in that respect. I think in the UK, we tried to sort of steering something a little bit different to the EU, but at the moment, it's not that different. There was this really exciting act that I think was brought in, Precision Breeding Act, which was to say that we wouldn't consider something a GM if it contained a variety of mutations that were the sort of mutations that might happen naturally anyway, and then you could just use traditional breeding methods through mutants to then eventually get to that. That was passed by all of the various parties, but unfortunately with the election just called, the legislation won't go through and it'll have to be moved till later in the year or next year. I think the one thing that was a bit sad about that act is it only covered plants in the end. And I think they missed a real trick there, especially with the interest in plant-based or non-meat-based foods, that they could also include fungal kingdom as well, which a lot of the foods, such as corn, a classic vegan food, is from fungal sources instead. It'd be great if they included microbes in that as well, but we'll see.
So we've talked about biopharma, we talked about alternative proteins, cultivated meat, a little bit about environmental sensing. Do you see any other fields where engineering biology could have an impact in the near future? Somewhere where we could see translation that might actually reach society?
I mean, I'll just jump in here first, sorry.
Yeah, please.
I came back from the big SynBio conference this week in the US, and the amount of groups that were no longer doing something to do with E. coli or yeast and metabolic engineering. So many of the presentations were like biosensing in soil, biosensing in wastewater, sensing on plants, right? So it's definite, real good application. I think if we go back to one of the original questions, what has happened in the last 5 years, I think one thing that's been amazing in synthetic biology in the last 5 years is the diversification into non-model organisms, thinking we can't just engineer E. coli to be the Swiss Army knife and tackle everything. It's going to be better if we find organisms that we can engineer that are experts at living in certain niches and doing certain things. And I think sensing is something really interesting that you can do, sensing and recording the environment in all sorts of places.
Yes, and probably a combination with what Claudia was talking about before, right? So if we can sense, can we also respond in the environment where we see a pollutant or whatever it may be that we're trying to detect?
Yeah.
Gogo, how about you? What other fields beyond cultivated meat?
Sure, I'll go a little bit into my own background because I see a bit of a connection there. This also connects a little bit with the talk that Claudia did less than an hour ago. I see a certain potential to, and I've always been very interested in the ability to mimic what is happening in nature, because nature has obviously been evolving for millions of years and has created some incredible organisms that are able to do things that are difficult for us to replicate in synthetic systems, but with the right engineering tools that we may be able to achieve things that perhaps haven't been able to be achieved before, but would potentially be quite valuable. The very classic example of this that I really like to go to is the electric eel, which is something that is capable of creating a huge amount of electricity within its biological system, even though, this is something that, perhaps from just a naive point of view, you'd think, oh, it's just a biological organism. How could it be so powerful that it's, generating a kilowatt of energy from just its cells, or our cells? Obviously electrical potentials in our cells drive a lot of function within the body, but we're not, shocking things when we touch them and, defending ourselves in that way. So I really like the idea, and I'm not going to say this is particularly practical at this stage, but I very much like the idea of using synthetic biology and creating mimic protocells that are capable of doing what cells within biological organisms like this are capable of doing. And a lot of this, in the case of the electric eel, is using pumps and across the membrane to establish these potentials, and using a certain geometry in order to create something that's greater than the sum of its parts, let's say. And I think And experts in this field might say this is crazy, but I would like to think that with some amount of effort, you could create a very similar type of system using individual cells that are synthetically engineered. And that could be something that you could use to create electrical system, an energy storage system that could be compatible with the human body in ways that you don't necessarily want to put a lithium-ion battery into your own body in order to power something like a pacemaker, but something that's synthetically engineered, you could easily imagine that. I think it's same for drug delivery. Sorry, go ahead.
I'm glad you weren't going down the route of electric shocking people.
Yeah, that's turning it into a comic book film, I think.
There's an author on my street who wrote a book where people gain the ability to electric shock people through natural reasons. Became some Amazon series, I think.
If you want to do that, the research, the space is wide open. So
it'll be fun.
Don't hold back.
Actually, that's a good cue for our final question for this initial discussion, which is, what advice do you have for the younger members of the audience that are perhaps doing their masters or at the early stages of their PhDs, considering a career in engineering biology?
Yeah, absolutely. So from my own context, I was finishing up my PhD about 4 years ago. And at that time, I was working, I was doing a PhD in physics. So it was quite removed, like I sort of alluded to at the beginning of the talk, from the engineering biology and synthetic biology type work that I'm now a little bit more interfacing with. So I think, even if, obviously everybody here is probably quite interested in this topic, but even if you weren't, it's quite easy to get into these things if you're opportunistic enough and open-minded enough to find and look for the opportunities that present themselves. Oftentimes this can be through looking at either academics or industrial companies that are working in spaces that you're interested in. Obviously, events like this where you can network and talk to people who are involved in these types of work are very helpful with that kind of approach. The internet is your friend. There's incredible resources, and also, people who work in at least, companies like ours, they like it when they're approached because it shows a certain amount of proactivity. You're not just sitting back and waiting. You're trying to, perhaps what you're looking for, and you're open to going for it and starting a conversation. And I think that if you do that, and if you're open-minded and willing to, jump into something that's maybe not exactly the perfect fit, but something where you think you can grow and learn and develop, I think that will give you a very good shot at moving forward. And this isn't always a straight path. You might feel like synthetic biology is your main dream and you'd only want to do that, but I suggest, at least in my case, I think it's been very beneficial to me to be very open-minded, not be tied down to exactly what I've done before. And in the end of the day, when you collect that large number of skills, you can generally become a very well-rounded person who has a lot of context and the ability to solve lots of different type of problems.
What are your thoughts, Tom?
Okay, well, yeah, I've been training people for nearly 15 years right here. So I got two. One is a really practical one for people doing research in this space and maybe beginning a PhD or something like that in synthetic biology or engineering biology. And that will be like that. Every time you do an experiment, you've got your tube of cells or something. And so the thing to remember is the unit of biology, the unit of your experiment is its cell, right? It's one cell. And it's basically impossible to just work on one cell. So you're always going to be working at least with several thousand cells. So you should be thinking about whatever experiments you're doing, try to be thinking about doing 1,000 or 10,000 experiments at the same time and come up with some smart way with barcoding and DNA analysis or something like that, so that every time you use that tube, you create as much data as possible. So you don't try to go for one design at a time. So we call that kind of combinatorial or multiplex experiments. And I think that's really become the trend now, and everyone should be thinking in that way. A less sort of practical one and a more sort of lifestyle one is try to be a connector. Like, don't think about, I'm going to join this research group and work under the supervision of this one person and that they're going to train me. It doesn't really work that way in real life. Like, the best people and the best projects come from going to multiple different research groups and the student themselves being proactive and saying, okay, I'm going to take the thing from this person and the technology from there, and they make the connections themselves. I think a great example of this I mean, we see it more and more in the UK systems of having rotations, but in the US system, it's very popular to have rotations. It's been the standard for a while, and a great example of this is this guy, Stanley, who's doing his PhD in the Bay Area, and in his PhD, he came up with this tool we all use called DCAS9. We had a whole talk about it from Kyla earlier, right, DCAS9. How did he come up with this? Well, his 3 supervisions in his first year were this person called Jennifer Doudna, who was working on this new thing, this guy called Wendell Lim, who was a synthetic biologist desperate for someone to come up with a way to control genes turning on and off in mammalian and yeast organisms, which was a difficult thing. And a final other guy called Adam Arkin, who was this systems biology symbio guru who knew about all these different things to do. And you can see a rotation of all of that, and it gives you DCAS9, which has become this tool for everyone. And so I think there's a lot of benefit there if you're a researcher on making sure you're sort of hanging around with multiple different groups and supervisors and bringing different things together from that.
Yeah, and I think the main message there was that all the supervisors were from slightly different disciplines, which is what engineering biology is about, because you're no longer talking about life sciences and engineering, you're talking about a combination and a fusion. And definitely, I think it is down to the students to create a fusion of knowledge and of approaches to solve something completely challenging in a novel way. Right, we're ready to open up to questions from the audience now. Yep, we'll just wait for the microphone so that people who are joining us online can hear the question.
Hello, thanks for the opportunity. So I wanted to ask about something that was mentioned before about large-scale AI models, particularly in synthetic genomics. And obviously these AI models are kind of sometimes referred to as black boxes, right? And I was wondering whether some kind of more mechanistic or mathematical models, more mechanistic-driven kind of theoretical input could be merged with this powerful kind of data-driven models by AI, and whether you see some kind of synergy where you can perhaps minimize the black box behavior of this or characteristic of these large-scale models with more so mechanistic or mathematical input.
Okay, maybe I could answer this. So there has been a lot of effort in the past 20 years to get mechanistic models that can help describe why certain DNA or RNA sequences, when put into cells, instruct things like gene expression, like how much transcription and how much translation there are, to be specific levels. And some of those have been really successful and highly used, but a lot of them, there's always a need to make the model more and more complex and feed it more and more data for it to be able to understand and also account for the edge cases. So things like sequences like hundreds of bases away that somehow have a big effect that you weren't taking into account. Now, more recently, obviously, there's things like neural network-based approaches, deep learning, which can take the big data sets and achieve the same thing with no mechanistic model considered. And actually, in many cases, they do it better than the ones with the mechanistic model. So there's a bit of a tension there of where we should go. Should we completely abandon the mechanistic model because it's just too much difficulty to think about it? And we are starting to see some researchers sort of go back and look to see how the learning worked and try to, the deep learning work, and try to sort of make interpretable models from that and bring it together. But at the moment, I'll be frank, everyone's a bit lazy, right? And if someone just comes along and says, oh, here's this thing, just feed it the data and it's better, then that's what people will do. And for me personally, I'm starting to think less as of DNA as something which we need to understand the mechanisms for, and thinking it more like just a foreign language that we just need to learn how it's written and how it's spoken. And actually, if you think in that way, then we don't really use mechanistic models that much anymore for translating one language to another. We just use these machine learning methods which can do it really, really well.
I guess I can offer an opposing view as a modeler. I would have to say that although I agree that there is information in the data that we may be missing the context of in a mechanistic sense. That's fine. I think the main hurdle is that quite often we don't collect information at multiple levels to a scale that would be large enough to give us insights into intermediate steps in a mechanistic network of events, right? And so if we were to generate that information, there is no reason why we could not have physics-informed data-driven models. So we can start building structures. And I think that is going to be key for correct interpretation, if I am honest. I'll give you one example. As I mentioned earlier, I work on glycosylation, and for a long time we've been looking at glycans as biomarkers for disease. Okay? Now, if you look at how glycans are made, we know the reaction mechanisms, and we know they are highly dependent. The structures are highly dependent. So if you see a particular structure, it needs to have gone through a processing pathway that is very well described. If you see less of it, it means another glycan structure has to emerge somewhere else. Okay? The other thing is that when we analyze through mass spectrometry, capillary electrophoresis, when we analyze this data, we assume that the protein that we're trying to analyze, every single protein has glycans, okay? So that creates another correlation that comes from the analysis, right? So we are talking about compositional data. If you go through the approach of building an ANN, and we have done this, we have proposed this, but if you go through the approach of building an ANN, there is no guarantee that the total sum is going to be 100%, which is what your assumption is, that you don't have macroheterogeneity. Or maybe you have quantified the macroheterogeneity. But the sum needs to be conserved, right? However, you can move to data-driven models that have some information about the network, which you know, so something like graphical neural networks, so a physics-informed approach that will guarantee that the results will always observe that sum being 100%. So I just think it's about getting enough data at every level to be able to interpret. And I think that is key for biomanufacturing, because as we go forward, so let's say we're making these de novo predictions about novel structures that can be used as therapeutics, but we need to make these structures, right? Several grams, kilograms of these structures potentially, or we need to make it on an individual patient basis, but we need to be able to produce it. And we need to know where the bottlenecks might be, especially when the structures are non-natural. Yeah. Next question from the audience? If there's any?
They're all so shy.
Yes.
I have a question that might be slightly off target for the science we're talking in here. But I have a question about funding in engineering biology. Of course, it's a big hot topic, and of course, it's going to cost a lot, and it's going to give even more. However, in terms of funding opportunities and where should we concentrate sort of with research, is it industry or academia, which is more sustainable and which is more profitable? Thank you.
Who wants to go first on that?
I think you have to, because we're not in the business of making a profit here.
Yeah, that's true. I would say it's fair to say that industry is more profitable in a certain sense, but I think they're just so, I certainly don't feel qualified to say that one is a better use of resources than the other. I think the very nice thing about academia is that you can invest the money to do the fundamental research without having it need to create a profit. The considerations that we deal with in industry are so focused on, especially for a young company, are so focused on how do we keep the company going? How do we make sure that it doesn't die off in the next round of fundraising? And there are these targets that you have to hit in order to be able to prove to people that you're making progress. And that can drive a lot of incredible productivity in a short amount of time, but it also doesn't always leave the room for as thorough exploration of all of the different considerations that are required for something this complex. And obviously, it has, based on all the things that we've been talking about, there's so many considerations that are involved in any of these topics. And within academia, you can have this a bit diversified where lots of people can be working on their own projects, they can be exploring things. Not all of them are going to be successful, many of them won't, or many of them won't go exactly the way that you wanted them to go, but you can still, as a sum, gain a lot of impact based on the findings that come out of this group or that group, or altogether it creates a body of research that is immensely valuable. And then I think what industry is very good at is translating something that's very specific and has shown a lot of promise into the change that you want that to create within the world, right? Sometimes academia will get stuck at a stage where it's like, okay, now we've got all of this findings, it's very interesting, what do we do with this? And you need then that focused body within industry to say like, okay, I'm gonna push this forward and make this into the product that people are actually going to use in the real world. So sorry if it doesn't very well answer your question in a very specific way, but these are my thoughts on that topic.
I'll say, I know all universities in the UK are non-profit, but it's well established that I think for every pound invested into research in academia, then it's worth about £3 or £4 back for the UK economy. In terms of driving innovation, attracting talent here, getting companies here. So it's always a good investment to invest in research universities, and of course I'm going to say that, and so we always want more money, so yeah, give us more money. But there is also a lot of debate at the moment in engineering biology where the UK should prioritize investment. The current government, and hopefully the next government, have talked about 200 million a year investment over the next 10 years to try and drive the bioeconomy. And I see a big problem in the UK and in most other countries in this space is a lot of what engineering biology can offer is moving existing industries into instead using biology to make the same products or better versions of the same products. Now, if you invest in biotech, you might know that maybe if you have a lot of money and you invest that spread over 10 biotech companies making drugs or things to go into the clinic, 9 of them will fail. One of them will get to phase 2, get bought by a big pharma, and you'll get all of your money back, right? And when I say a big pharma, I mean a pharma company, not like a really big guy in a field. But then there's all these other applications with engineering biology where it might be like a guy in a field has to take it on, right? And so if you think about one great thing you might do with engineered biology, it might be get bugs, microbes to ferment and produce the precursors to some of the more complex and expensive plastics that instead we have to make through really polluting ways from petrochemicals. The UK has one of the biggest plastic companies in the world called INEOS. But I don't see there being a way where if I invest in 10 microbial kind of plastic monomer making companies, one of them will eventually get to the point where INEOS buys it and puts it into their factory, because their factory is for completely different infrastructure. It's for doing stuff with crude oil. So I think a good investment for the UK will be to think where those gaps are at the sort of larger infrastructure scale, so that we can get companies that want to ferment products that aren't for medical reasons, which often only need to be made in small amounts, but instead things that are going to be in much bigger amounts. We can sort of reduce the risk for those kind of companies.
And we can prioritize certainly looking at sustainability, right? That's
Yeah,
exactly
definitely.
what Tom said without saying it explicitly, that as we move away from these oil and gas derivatives, we need to think of a sustainable way to really scale up engineering biology innovations and make commodity chemicals, which we don't really do.
Yeah, and one thing I've noticed in the UK as well, which I think is really good here, is that there is quite a lot of venture funding at the early stage. That is, these guys running the pots, they can only spend it on sustainability. right? And so they're really driving them to think away from just like, okay, let's just invest in another checkpoint inhibitor drug or something like that. And they're instead looking at companies like Maltus and all sorts of companies in all sorts of diverse spaces like bioconcrete and reduced CO2 emissions, left, right, and center. It's just finding a way to get those ones to then be able to scale when then they can really make the difference. I think that's important.
Yeah, and to have the infrastructure to do these studies, these scale-up studies, which I think the UK is largely missing. There are some alternatives in continental Europe.
Okay.
But I think this is something that the government is looking into. What sort of facilities do we need? We currently only have CPI as a center of excellence. So how do we build regional hubs that can help us? Give us the right advice to scale up innovations that come out of universities. All right, I think there was one last question.
So engineering biology is very tool-driven. And so, 10 years ago, if you wanted to knock out a gene, you could use talons and put a lot of postdoc time into it. And if you wanted to sequence the entire genome of something, throw a few hundred postdocs at it, you can sequence it. Now we can do it a lot quicker. So if you have, rather than infinite numbers of postdocs to throw at a problem, we'll develop the tools to come up with that. What are the actual things that you can't do with an infinite number of postdocs? What is the tool that will be a complete step change in engineering biology that will allow us to do stuff that isn't just a resource constraint, it is just a fundamental, we can't do this now, but want to?
I'll answer this from a slightly different perspective. I was asked, why isn't engineering biology as easy as AI? At a House of Lords inquiry recently, and the answer is because if you want to do a startup in AI, where do you begin? You open up your laptop, and then you buy compute from Amazon Web Services or whoever, right? And instead, this ginormous infrastructure of some factory somewhere and some factory somewhere else, they've built the laptop, semiconductor factories at insane costs, server farms somewhere, who knows where, like, is it in the east of Washington State? All of Amazon's server farms. You've got this humongous amount of multi-billion infrastructure that does it all for you, right? And then so that you're just the one doing the coding and running it. That's what I would like to get to with biology, that we would have these sort of cloud labs, cloud, everything. Like, it wouldn't be like, let's just go and design the DNA and this small amount of DNA comes and then we have to work out what to do with it, right? What if we could design the DNA and then it gets built somewhere and it gets tested somewhere and we get the data and it gets done and we just get the data back? Right, that's the kind of infrastructure that would be game-changing, but it was going to take a lot of money and a lot of work to get to that.
Great. This is all we had time for today. Thank you very much for being here and for asking questions. We will be back at 4:30 sharp for the annual lecture of this event, which is going to be delivered by Kristalla Prather from MIT. So see you back here in 10 minutes.
works. It's like, it's not.
Okay, welcome everyone. We're ready to start. Okay. Hi everyone. Welcome those of you that join us for this, the highlight of our day. We've had a very exciting day discussing engineering biology. Many of you were here before, and as I said, those that join us now, you are very welcome to be here. I am Amparo Galindo. I'm a professor of physical chemistry in the Department of Chemical Engineering, and I'm also the director of IMSE, the Institute for Molecular Science and Engineering. Before we start with the lecture today, I have a little bit of housekeeping. As you see, we are very lucky to have a British Sign Language interpreter for our lecture today. There will be, in fact, 2, so there will be another one arriving. If any of you need the interpreter, please move to the front if you haven't done so yet. And because the content of our talks is quite or very technical, and some of us speak also very quickly, people like me, the interpreters will switch every 10 minutes, so you will see them switching. So yeah, I hope you're not too distracted by that. I hope especially our speaker will not be too distracted. We are very lucky, as I said, to be able to have an interpreter today. So let me briefly just introduce today's talk. Let me tell you a little bit. We are, as I was saying, the Institute for Molecular Science and Engineering. We are one of the Grand Challenge Institutes of the college. We work with a remit to facilitate and promote interdisciplinary work throughout the college. We draw on the strength of our 4 faculties, and in particular in IMSE, we are interested in problems in which molecular innovation can deliver solutions at engineering level or at function level. So that's really the work that we do. And our event today on engineering biology is a prime example of how disciplines coming together lead to better solutions. And there's been plenty of examples of that today, and we will see more in the work of our invited speaker. So as I was saying, this is our highlight event of the year. This is our 6th Dr. Theo George Wilson annual lecture. This lecture is possible thanks to a very generous award by Theo Wilson, who is a PhD alumnus of the Department of Chemistry. He graduated in 1952 here with a PhD and went on to study medicine in the US, and he now works in the US. And so as I was saying, he donated a very generous gift to IMSE in particular, and in quotes, at the time of his gift to us, he said that he wanted the money to play a part in furthering the tradition of great scientific advances by Imperial. And he said, I hope my gift will be of great benefit to students in the future. So as we made the decision of how to best use the money that he was gifting, we decided that starting this series of lectures to keep it as broad as possible and with areas of interest that can include as many of us as possible was a very good use of that money. And in addition, we created a scholarship every year to support an EMRES student working in the area of molecular engineering, because that is the EMRES that we support in the institute. Okay, and so with this, my other task is to introduce Professor Kristalla Jones-Prather. We are very privileged to have her here today with us. She is the Arthur D. Little Professor of Chemical Engineering in MIT. She's also the current head of MIT Chemical Engineering. In terms of her background, she obtained her Bachelor of Science in MIT in '94 and did a PhD at the University of California, Berkeley, and graduated in '99. From her PhD, she went on to work in R&D for Merck for 4 years. So it's, again, a very interesting bio, moving from academia to industry and back into academia, which, again, very, very successful, and, showing how taking, or experiencing different environments can lead to very successful careers. In terms of her work, I got some information from her webpage. So she describes her work as engineering microbes to produce chemical compounds. And I think we're going to hear some of that today. And she's interested in metabolic engineering, biochemical engineering, bioprocess engineering, and synthetic biology. Kristalla, as I was saying, we're very honored to have her here. She has a long list of awards. I selected a few. There were pages. So I have just highlighted some of maybe the most impressive one, also the recent ones. But I leave them there for you to read. I'm not going to read all of them. What is, I think, exemplary of the career that Cristala has is that there's there a list of awards that go from technical awards, like the AICHE award at the top, to also plenty of teaching awards, and also advocacy awards. I know that you're a big advocate for inclusion at work, for equity, which is something that is very strong for us in IMSE in particular and in college in general. So, really very happy to have you here, as I was saying. And so with this, Cristala, I'm going to give you the floor. Thank you so much for being here. She's going to tell us about building microbial factories, design, assembly, and engineering of biological routes to chemical compounds. Please welcome Cristala with me.
Well, good afternoon, everyone. It is a pleasure to be here. I have to give a warning up front, which is that my task was to present a talk that would be accessible to a wide audience. And so I did what many academics do, which is choose to talk about a lot of stuff. With the hopes being that if you don't understand the first part, maybe you'll understand the second part. If you don't understand the second part, maybe you'll understand the third part. If you understand none of it, maybe I'll tell a joke or two that you'll enjoy along the way. And if you still don't like my jokes, hopefully you'll enjoy the reception afterwards. So I do, all the work in our lab is really focused on this idea of engineering microbial systems for chemical compounds. But we're motivated to do this with a very specific purpose. And I use this graphic as an illustration of how we think about where the material comes from. I'm a chemical engineering by training, and I often tell chemical engineers when I'm teaching them in our introductory class that chemical engineering is about stuff. It's about making stuff, it's about characterizing stuff. It's really about working with materials and molecules all around us. And so a natural question for me then is, where does that stuff come from? And the reality is that a large amount of the stuff that we need and that we use comes from petroleum, comes from non-renewable sources. As a chemical engineer, I'm also a person who teaches in this intro class about conservation of mass. And so if we believe that fossil fuels are actually the byproducts of fossils, then it can only be that we are consuming them at much faster rates than we're producing them. So we have a fundamental problem here. Now, the part of this picture in terms of where materials or stuff comes from that we're most accustomed to thinking about is the largest fraction. And you can see the numbers that are there are in gallons. Most of it is about energy. And there are lots of ways that we can think about displacing that mass for energy. And the example that's shown here is that of an electric vehicle. So when we think about taking the carbon that's in a barrel of oil and asking how do we power mobility or how do we power devices, there are many ways that we can think of to address that don't require the utilization of carbon. However, there's a smaller fraction of this, but a fraction that, from my perspective, is just as important, that's not about the carbon mass being used for energy, but the mass being used for mass. That is, it's about making the stuff. And the graphic that's shown there is an indication of all of the different materials, representations, I should say, of the different materials and the different places in which this carbon that comes from petroleum or from fossil materials is used for things that are very important to us. It includes the plastic and this clicker in my hand, the materials of the chair that cover the chairs that you're sitting in as well. So when we ask ourselves, how do we actually think about replacing that stuff? And we think about where does this come from? We start from crude oil going into fuels and also into chemicals. And then ask, well, if we can't keep relying upon this resource that's already there, where do we get access to carbon now, mass for mass purposes, that we could have on a regular basis, on a sustainable, renewable basis? And that leads us to biomass. So the title of the slide that you can see here is describing a paradigm shift. I've borrowed this from one of my colleagues at MIT who talks about the need to go from being based on hydrocarbons to carbohydrates or in other words, from being based on fossil materials to being based on renewable biomass. What that means is that we need a way to think about how do we take those materials now, this biomass, and can convert that. It could be converted into fuels, and especially these days, the focus on aviation fuel. I've gone on record as saying I will not fly a battery-powered airplane to the UK from the US, right? So that's an area where we have to think about, because of the density there, about replacing mass with mass. But it really is this much wider array of biochemicals and biomaterials that's attracted our attention and motivates the work that we do. Now, as a chemical engineer, this was the view I was given of a biological cell when I was a student about 35 years ago, which is, it's this thing, don't worry about it, it's just a cell. What we know, of course, is that a cell is comprised of many different reactions and many different metabolites. And I liked very much earlier today, Mark Howarth said, biology is the search for chemistry that works. Okay? And in effect, biology is chemistry. It's a collection of all of these reactions that give us form and function. And we've been able to show that you can take simple inputs like sugar that can come from biomass. In some cases, you need to provide oxygen as well, and other nutrients, those components that give us biomass. And from that, we have a rich history of producing, especially pharmaceuticals. So being able to have natural products that have therapeutic functions to be able to address different diseases. And you can see the representative molecules that are shown there, as well as the diseases they were indicated to treat. But we've also been able to use biology to produce things like amino acids, which are natural products in biological systems, and also to produce different kinds of organic acids, some of which are natural to biological systems and some of which are not. So this tells us that biology already gives us a tremendous reservoir or a tremendous set of molecules that we can choose from, but more importantly, a tremendous set of reactions that we can choose from in order to build new ways of using biology to build chemical compounds. So if we go back to the same diagram I had before, what we're really doing is taking a very simple molecule, which shown here is glucose, and we have to traverse this very complicated network in order to make a particular compound of interest. And I'll come back to this compound that's shown on the slide in a minute. As we do this, one thing we're concerned about are byproducts. In other words, what are the compounds that are made that we would prefer that the cell not make? Well, one of the things that we know is if the cell is making this compound, it must have come from a reaction in that cell. The reactions are almost all mediated by proteins, specialized proteins called enzymes. Those proteins are encoded by DNA. And so if I know where the unwanted compounds are coming from, and I can identify the protein that makes that unwanted compound, I can remove it. And we heard many talks today about ways in which you could remove them, including ways if you want to string too many things together, you may run into trouble constructing these systems to be able to do that. And this is very classic metabolic engineering. Both Kyler and Karen's talks this morning talked about pathway engineering. But one of the challenges that we've asked is what happens when what you're competing against is not some other compound that's being made, but rather it's the desire or the need of the cell itself to grow. In this case, you don't have the choice to simply knock out growth or eliminate growth, because if the cells don't grow, they don't make these proteins, the specialized proteins that are enzymes, that you need to do the chemical reactions to make the compound that you want. So we're faced with the particular challenge here, and how do we actually address our desire to funnel or to channel as much of the carbon that we provide into the specific compound that we want, while balancing the fact that the cells don't really care what we want, right? So we're working against their interests at all times. So with that as context, there are two primary objectives we've had for nearly all the projects we've had in my lab over the past 20 years. The first is to be able to increase what I'm referring to as the nodes in this network. So in this diagram that's shown here, each of these dots represents a chemical compound, so that's some molecule present in the cell, and then the lines between them represent chemical reactions. So when I say increasing the nodes or increasing those dots, what we're trying to do is to find more ways for biology to make chemical compounds and in particular to make compounds that are specified by us for particular uses. And then the second thing that we focus on is how do we actually get enough of those compounds being produced to be significant, to be relevant, to be something that would actually be useful. So we have one part of our lab which focuses on how do we actually get new molecules, and then very quickly those projects turn into ways to try to understand how do we push the capacity of biology to do more and more of the chemistry that we want. So the rest of my talk, I'm going to walk through a series of examples of different targets that we've selected, what our particular challenges are, and hopefully you'll see a narrative that goes through in terms of general themes and approaches that we have to tackle this general problem. Okay, so I am going to start with the production of a compound called glucaric acid. There's a reference that's shown here on the bottom of the slide, and so you can see that we first started working on this a long time ago, but it has presented a number of challenges, and I always tell my students, challenges are just opportunities. So when you are struggling, remember, you have been given an opportunity to overcome something very difficult. I won't spend a lot of time talking about how we developed this pathway because, again, we generated it some time ago. But I'll just say we actually started from the end and worked our way forward, meaning we started by asking where does a compound like glucaric acid come from? It turns out this is a natural product, and we identified a bacterial source of a protein that could make that compound from a substrate called glucuronic acid. If we asked where glucuronic acid came from, we identified a mouse version of a protein to make glucuronic acid. And if we asked where we could get myoinositol, the precursor for glucuronic acid, from, there had been a study already published that had shown that that compound could come from only adding one gene to E. coli, and that was the gene encoding a protein that came from Saccharomyces yeast. So this was, in concept, a very simple pathway. It has 4 reaction steps, but it only requires 3 new proteins to be added to the cell. But the challenge that we have is that the first intermediate in this pathway is a compound called glucose 6-phosphate. How many of you have taken a biochemistry class? How many of you are getting warm, fuzzy feelings thinking about biochemistry? There were a lot fewer hands with the second question than the first one, okay? But biochemistry can be useful. And those of you who are thinking fondly about biochemistry are immediately going, yes, I see the problem. Glucose 6-phosphate is the first committed step for glucose metabolism in E. coli. Meaning what the cell really wants to do is take that glucose 6-phosphate and use it for its own purposes and not to make this compound glucaric acid that we're interested in. And just very briefly, here's an overview of the different reaction steps that happen in E. coli when it, meaning when the cell, wants to use glucose to make more of itself. And what you'll see here is that we have the primary glycolytic pathway, we have the pentose phosphate pathway, there's a minor reaction to make a small amount of glucose 1-phosphate. I'm not going to go into a lot of details here, I'm just going to say that our goal is to be able to take glucose 6-phosphate and have more of that be available to make the compound that we're interested in. We use both what was available in the literature and some of our own modeling work to identify the ZWIF protein, or glucose 6-phosphate dehydrogenase, as something that we could eliminate from the cell. That way now we know that the glucose is mostly going down that pathway shown in the middle. And then we also identified one particular enzyme in the middle as the point where if we could control the ability of glucose to go through that particular point, we could regulate whether or not it was being used for the cell to grow. And you can see where biomass is indicated on the slide as something as indicated of the cell growing, or to be able to have what we call flux, or essentially have flow through that point be restricted such that we have more of that substrate available. And if we have more of it available, we have more of our first intermediate and ultimately more of our glucaric acid being produced. So conceptually, we knew what it was that we wanted to do, and then the question was how? What is the mechanism? What are the specific pieces of proteins or DNA that we're going to assemble together into a cell to be able to build what we call a metabolite valve? We call them valves because if you have ever turned on a water faucet, that faucet has a valve in it that allows, when you open it, for the water to flow, and when you close it, for the water to not flow. In our case, we want a valve that when it is open, it allows the glucose to be used for growth, and when we close it, and we don't close it all the way, but when we close it just a little bit, there's less of it available for growth, and more of it is available to go into the pathway that we're interested in. So we actually demonstrated a couple of different ways of controlling this. I'm going to give you an example of one that has been a theme we've carried throughout other work in the lab, and that is using something called quorum sensing. And I'm going to pause and go through this in a little bit of detail, because if you've heard of quorum sensing, it makes perfect sense, and you don't need any explanation at all. If you haven't heard of quorum sensing, it makes no sense. And I can give you a little bit of a foundation to help understand what it is that we're trying to do. So I'll turn to this one to make it a little bit easier. Our quorum sensing system has a couple of key features. One of them is a special protein, a regulatory protein. You can see it here as ISA-R, whose job it is to sit on DNA. And it will bind to a specific sequence of DNA and will sit there. A second component is what we call a ligand or a signaling molecule, which is a small molecule that will interact with that regulatory protein. And when the molecule is absent, the protein sits on the DNA. When the molecule is present, past a certain point, the interaction of that molecule with the protein causes it to come off of the DNA. In this particular case, we need the DNA to be open in order for us to make a protein. So DNA goes to RNA, RNA goes to protein. So if I don't have any of this signaling molecule, my regulatory protein sits on the DNA and it prevents me from making the protein of interest. If, however, I have enough of this ligand accumulate, then the protein comes off of the DNA. That allows then for another specialized protein called an RNA polymerase to come in and convert the DNA into RNA, and that allows me to have protein. Now, the last component I'll mention here, so I've talked about the places where the regulator binds. Those are our promoters. The regulatory protein I've mentioned. This last component is called an AHL synthase, and the AHL is the signaling molecule or the ligand. That is the small molecule that binds to the regulatory protein. There's another protein in the cell that produces that molecule. And so with those key components, what you have now is a cell that will produce the signaling molecule. At low amounts, it leaves the cell and it just floats around in the space where the cell is being cultivated. When it reaches a certain concentration, it will come back into the cell, and when it comes back in, it now is able to interact with this regulatory protein in a way that it can cause it to be displaced. Okay, so why is it called quorum sensing? Well, typically we think of a quorum as being a sufficient number of people or sufficient number of items for some action to take place. If I now have this signaling molecule being made by the cells, the more cells I have, the more quickly I will make that signaling molecule. And there's some critical amount of the cells that corresponds to a critical amount of that signaling molecule to trigger the reactions that we see. Hence, if the cells establish a quorum, then they're able to trigger gene expression. The benefit of this to us is that it allows us to construct a system where what we're actually doing is allowing the cells, based on how quickly they grow and how quickly they make this signaling molecule, decide when they want to switch our valve. This diagram, the way I've shown it, has the cells starting where the protein to be controlled is off and is turned on. But the particular system that we use, and the reference for that is shown here, and it's named here, actually has a different mode where that same regulatory protein can bind to a different sequence of DNA located a little bit further ahead of where the first site is. And now when it binds, it actually brings with it the protein that we need in order to make our target. The same interaction happens where as that quorum sensing molecule is built up, the regulatory protein is displaced, but this time when it comes off of the DNA, it takes the RNA polymerase with it. So now we have a configuration where we start on and we're turned off. So this works great for the case I described, where we want to start by letting the cells grow to make all the proteins we need to trigger our reactions. And then at a certain point, we want to say, you've grown enough, you've been selfish enough with our carbon, and we're going to have you stop growing and then utilize the carbon that we're providing in order to now have our product of interest being produced. So we can vary the promoter to go from on to off, and then we don't know exactly when is the right time for switching, but we can actually build a library that expresses or makes that signaling molecule at different rates, and then just screen the library for the best time for the cells to switch. Okay, so that's an overview of quorum sensing, but I want you to take something else from this slide, and that is that in general, we can control whether or not a protein is being produced by being able to control whether or not a regulatory protein is sitting on that DNA or coming off of the DNA. And if we have multiple choices for how a regulatory protein can be displaced from the DNA, then we have multiple ways that we can control the flow of metabolite and therefore control the production of compounds. Okay, so I'm just going to show one result of having gone through this whole process of now looking for the right point to be able to switch between producing cell mass and producing a product of interest. And in this particular case, we actually wanted to make sure that the system that we developed would work at different scales. So we had a panel discussion right before this talk where one of the questions that was asked was about translation and how do we actually consider translation and going from academic research into what's happening in a company. And it's something that we think about a lot as well. So we want to make sure when we're building new systems, new tools, that we've tested them, not just in the smaller scale systems that are useful for being in a research lab, but also in these bioreactors, which give us a much better indication of how it might perform if we go into manufacturing. So the standard that we use is actually a shake flask. So that's a 50 milliliter culture volume and a 250 milliliter vessel. There are 4 different samples that are shown here on this chart. To the far right is a wild type. So this is a cell that doesn't switch at all. It has the pathway, meaning it has the genes that it needs to make our product of interest. But you can actually see under these circumstances for these cells, they don't make the product. The product is shown in the blue bars here as glucaric acid, and the yellow bars are one of those byproducts. And we had actually already tried what the typical method is, which is to knock out the genes encoding, in this case for acetate, but that gave us a cell that wouldn't grow at all. Okay? So we were actually faced directly with this problem I presented, which is what if you can't knock out the pathway for the thing that is your undesired byproduct? But in this case, it was both growth and an unwanted acetate molecule. The 3 other strains that are shown here are 3 different strains that will switch at different times based on the rates at which they are making this signaling molecule. And so we have something that switches very late, so it looks a lot like wild type. It makes no product of interest, only acetate. We have one that switches pretty early, in which case it makes some product, but it can't quite figure out what to do, and it makes a lot of acetate. And then we have what's our Goldilocks strain, which switches just at the right time, and it accumulates no acetate, and it makes only product of interest. We could test this on a scaled down model. I actually saw one of these at White City yesterday called a biolector, where you're using 1 milliliter culture now, so 50-fold smaller than the scale in the center. And you can see that both qualitatively and quantitatively, we're achieving about the same amount of product being produced. And then the next step was to look at how this might scale up into a larger reactor. Okay, how many PhD students are in the room? Okay, I'm going to tell you a story, and you can't tell your advisors. Okay. So at this point, the student who was working on this project came to me and said, I don't want to do the experiment. I think that's a phrase that has never been spoken by any of you who raised your hands. It's a unique phenomenon in the United States. And then I said, so tell me why you don't want to do the experiment. And he said, well, in these strains here that are making a lot of acetate, I think that's causing the pH to go down. Yeah, it's an acid, right? You make a lot of acid, the pH is going to go down. And then he says, well, I think it's the low pH that's actually causing the cells to not be able to make product, because we know that if the pH is too low, you actually have trouble transporting glucose into the cell. Okay. And then he says, well, if we're in a bioreactor, we can control the pH. And if we control the pH, I don't think the system is going to perform quite as well as it otherwise would. Therefore, I don't want to do the experiment. So you have a conversation that goes like this. There's no such thing as good data or bad data. Your reaction to the data might be good. Your reaction to the data might be bad, right? But we're here as a search for truth, right? In other words, go do the experiment. Okay? And he was kind of right. So what happened when we had the large-scale reactor is that the wild-type strain, so this is shown here, did make a lot of acetate. But E. coli can re-assimilate acetate. It can take it back up. And with the pH being neutralized, that's exactly what it did. It made a lot of acetate, then it took that acetate right back up, and then it could make product. However, this actually had a much lower titer than the case where we were controlling it much more directly. So we had more biomass, less product in that case. In the case where we had our valve, we accumulated no acetate, less biomass, more product. So the key take home here, one is that we actually were able to show the consistency across scales, which is very nice. And then the other, I borrowed this line from my good friend Tony Sinsky at MIT, who says the best metabolic engineering projects are ones where you start from 0 and make anything, because now you get to divide by 0 and say that you have infinitely improved the performance of the system. Okay, that may be a stretch, but what it really showed us is that we took a case where, for this particular environment, for these strains, we had trouble producing a compound, and then our ability to actually regulate the flux of glucose to keep it from going into endogenous metabolism kept us from accumulating a lot of acetate and allowed us to make our product of interest. Now, I mentioned that this was a pathway we've worked on for a long time because it's given us a number of challenges, and those challenges are opportunities. And I want to tell you very briefly about a different step in the pathway, and this is shown here where we use this mouse protein to go from a compound called myoinositol to glucuronic acid. And we knew from our first publication that the activity of this protein was very odd. It was much higher if you synthesized the protein when its substrate was present. We also found that the activity was highest early on, and it dropped off much more rapidly than the other enzymes in the pathway. It's not unusual for an enzyme to be highly active initially, and then after a few days, the activity to go away. But this one, the activity went away very, very quickly. So that actually gave us an idea that if we could delay the expression of that enzyme until its substrate was present, then we would perhaps see higher titers. And I'm showing here a graph where we did that experiment, where we had 2 different enzymes, or 2 different genes. We controlled then the expression of those 2 different genes to make the enzymes with 2 different inducers. And if we delayed expression of the second one relative to the first one, you can see we got higher products being produced. So that was nice, but again, if we're thinking more towards translation, we don't want to have an operator go into the system and decide, now I want to induce this second protein. Instead, what we would like is for the cells to be able to say, okay, I need to be able to turn on expression of this protein now because this molecule that I need to be present is there. So if you remember when I described quorum sensing to you, I said the key features were a protein that binds to the DNA, and then something else, in that case it was this AHL, that when it is there, allows for the protein to come off of the DNA. Well, it turns out there is something exactly like that that responds to the substrate for this reaction, which is myoinositol. It's a protein found in a completely different microbe than E. coli. It's in an organism called Corynebacterium glutamicum, and it works exactly like the system of quorum sensing works, where if the molecule it responds to is not present, it sits on the DNA, and it sits at a spot that we choose, and we chose a spot so that it would turn off gene expression. And then when now the product of interest is there, what we're able to do is to have that myoinositol give us the ability to turn on expression of that gene. These we now call biosensors. Okay? And just roughly speaking, a biosensor is anything that will respond to a particular molecule. In this case, they are biological molecules, they are proteins, and they're responding to metabolites of interest. And so what we wanted to do was to build then a biosensor that will allow the cell to decide when is the right time to turn on this particular protein. I just want to show you how we build these biosensors. I'm going to show you several sensors coming up later where I'm not going to talk about how we build them. But one key feature here is that we know if this protein sitting on the DNA has to be displaced by this new molecule coming in, this ligand, then how much of that ligand we need to displace it is going to depend upon how much of the protein we have. So what I'm showing here in these 5 different colors are 5 different samples where the regulatory protein is being expressed either at very high levels, that's the lowest, or at lower levels, which is the one at the top. And the line that's fairly straight across the top is the case where we're not regulating anything. So we just have, in this case, a GFP reporter being turned on all the time. So you can see if we don't make a whole lot of that regulatory protein, then it doesn't take a lot of the ligand being added before it can give maximal expression. But if we have too much of that protein, we actually can't add enough of the ligand in order to have it be displaced. Okay? But in all cases, what you see is that we are getting expression of this protein as we add more of the myoinositol, and we can sort of tune how much we make based on how much of that ligand is present. That experiment was done if we just added the myoinositol to the system. We now want to see what happens if we're making it inside the cell, because that's the use case that we're interested in. And what we see here, first the sensor responds in a dose-dependent manner. If we're now looking at what happens when we're making this compound in the cell or not making it in the cell, you see the same sort of reaction, that where the lowest activity is there of the promoter, we get the highest signal from making the compound inside the cell, and where we have more of it, we have less. The next part then is to actually use this system to directly control the expression of the production, rather, the compound that we're interested in. The no IPSA case is the case where we can't turn it off and it's on all the time. And then we have these 5 different cases, and we don't know exactly when is the right time for gene expression to be triggered, but we can let the cells choose. And you can see there's one case where we actually have this expression being controlled by myoinositol, and it results in an increased expression of increased production of the compound that we're trying to make. So the last slide that I'll show is that the first case I told you, or showed you rather, of being able to control the amount of glucose going through the cell. In this particular case, even though they're on the same pathway, they have 2 totally different mechanisms. So we can put them together, and what was before a 2 to 3-fold increase in improvement for either one of them by themselves actually became about a 10-fold improvement when both of them were put together. We have used this general approach of saying, okay, how do we control the flow of material, both to continue to look at new quorum sensing systems. I'm not going to talk about that today, but we also have been inspired to think about new ways of using biosensors, that is, using ways for pathways to be controlled based on particular small molecules in a number of different applications. And so I want to introduce, I forgot about this slide. I told you it was a new talk. Okay, so let me come back to glucaric acid and this compound that we're producing. And I want to tell you why we're making it. We originally chose it because it was on a list of compounds that from a report of the US Department of Energy, there was a similar report that came out from the EU a few years after that. The first report came out in 2004. The next one came out around 2010 that identified this molecule, glucaric acid, as an interesting compound. And one of the things that they pointed to had to do with the efficiency of cooling towers. Okay? Now, they didn't talk specifically about cooling towers. What they talked about was water treatment and water usage. And in fact, what was known was that phosphates were the primary molecule that you would add to water treatment products to be able to chelate heavy metals in industry. The problem with phosphate is that if you have runoff into natural waterways that are normally phosphate limited, algae that exist in those water bodies will now see a lot of phosphate. They have plenty of sunshine. They will grow. As they grow, they block out the light for the aquatic wildlife underneath. So you have a process known as eutrophication, or essentially as an algal bloom, and that actually will kill the fish that are underneath it. Well, what we knew from that study and have since been able to show, and this is Callion as a startup I'm a co-founder of, what we've been able to show working with partners there is that glucuric acid is a great replacement for phosphates. Again, that was actually known for the report. What we didn't know until recently is that actually using glucuric acid in place of phosphate for the water that's treated and goes back into these cooling towers actually cuts down on fouling. So the chemical engineers in the room are now thinking, yes, this is my thing. She's talking about heat exchangers and cooling towers. I'm still a chemical engineer at heart. The biologists have no idea what I'm talking about, by the way. So if you actually reduce the fouling, and fouling is essentially a process where the inner lining of the pipes have metals that deposit on them. What that fouling does is actually reduce the efficiency of the cooling towers. And so we've actually done some studies, or some back-of-the-envelope calculations, I should say, based on some initial studies that show that not only can you have cleaner runoff if you replace phosphate with glucuric acid, but you can actually reduce CO2 emissions from industrial waste facilities by the equivalent of about 230 million metric tons per year. And that can result in cost savings of about $60 billion US to the need to be able to refurbish or to renovate or get those facilities back up again and cleared of all the fouling. So I had promised I would tell you what glucuric acid was used for, then I forgot that I had promised I would tell you that, and then the slide was there to remind you of it. So this is something that we're pushing forward with in partnerships to be able to bring cleaner solutions to be able to address the environmental demands for industries that we can't quite get rid of yet, but need to be much more efficient in their carbon footprint. Okay. Now I'm going to continue with the biosensor study, and there's a lot of details on these slides. In the interest of time, I'm not going to go through them in a lot of detail, but for those of you who are working in this space, if you want to ask me questions about it afterwards, I'm happy to go into a lot of detail. In this particular case, what we decided is that we wanted a biosensor to be used to help us control whether or not a pathway to make a particular compound was turned on or off. And again, I'm going to wait and give you the punchline for why we wanted to do this a little bit later. But I'll say in this case, we were interested in a very specific substrate or a very specific starting compound, which is called galacturonate or galacturonic acid. The substrate was actually more important to us than the product in this particular case. So based on the substrate that we had available, we actually started to look for what some interesting end products might be that would be useful to make from galacturonic acid. And we settled upon glycerate as a test study for that. And you can see to the far right as a reference that describes all the ways in which glycerate can be useful as an industrial chemical. Now, it turns out that E. coli cells have a way to use galacturonic acid, but in particular, if we introduced 2 new enzymes into the pathway, we could produce this intermediate that was already known in E. coli metabolism, and then the gray arrows here represent what the cells will already do. So if we introduce 2 steps, we could then turn the rest of it over to biology. Biology is a search for chemistry that works, right? We could use the chemistry that was already shown to work in E. coli to make this glycerate. So we developed a sensor for this in the same way that I described before. I'm showing here what those traces look like, but those are just different ways, as I had said previously, of having different strings of promoters. In this case, we also have different places where we've put the strings of DNA that the regulator binds to. But all these different versions give us a range of different constructs that we can use. And in this case, rather than letting the cell decide, we decided we wanted something that had a very high dynamic range. That means that if we take the maximum output and divide it by the minimum output, then we have a high change there. And we wanted a low leakiness, meaning that when our substrate is absent, we want the expression to be as low as possible. So I've highlighted there the particular construct that we chose. And in this case, it became very important that we find a way to control the pathway to make glycerate. And that's shown in this particular slide. So this is actually a set of growth curves that shows how the cells grow in the process of making this product of interest. And I want to point out on the far right, this gray line. That gray line is the cells growing if we're trying to make this pathway all the time, meaning it's always on, we're not controlling it at all. Those are very unhappy cells. Okay, so this is the growth as a function of time. It takes them a lot longer before they start growing. They eventually do get to the growth of the other strains, but they are not very happy. If, on the other hand, we let the cells grow without turning the genes on, and then we induce them, then the cells grow like this. And all these different traces show what happens if those genes are being turned on because we've added galacturonic acid, meaning we're using our sensor to do the work, or if we've added something completely separate, just a regular inducer you can use for almost anything called IPTG, and that's shown here as IPTG. So those cells are equally happy if we induce with something that's not associated with the pathway than they are if we induce with something that is associated with the pathway. And in fact, they actually produce the same amount of material if we are inducing with the substrate that's already going to be there, or if we're inducing with something else that we have to add. So what I'm showing here, this is now the substrate being consumed, this is the product being produced, and the peak bars for both substrate consumption and product formation are in the case where I have this other molecule, the IPTG, and the dark blue bars are what happens in the case where I let the cell decide to turn on the pathway when that substrate is present. As a note, these gray cells, which didn't grow very well, they also cannot consume much in the way of substrate, and they cannot make much in the way of product. Okay? So what we have here is a system where we know that inducible or controlled expression is required, and we've been able to show that we don't have to spend the money for an extra inducer. We can just let the cells respond to what we're already adding into the system and go from there. Okay, here's why we were interested in this. We were interested because we had a project from the US Army where we were presented with the following challenge. Army bases have a lot of food waste, okay? And the amount of food waste that you have is quite variable. And we all know about composting, and you can make biogas from composting, and composting works with pretty much anything. But the Army was interested in whether or not you could take a really heterogeneous mixture like this and use it to make something that wasn't a gas. Could you make a specific chemical compound that, for example, might go into a fuel cell and it can make electricity instead of making something else? The problem with that is this food waste is going to vary a lot. It's going to vary based on what was cooked one day versus the other. It may vary based on how good the cook was one day versus the other and how much people actually consume. But our challenge is, how do we take this very complicated mixture and get a very specific product that comes out of the end of it? Okay? And as you might imagine now, since I said I would tell you why galacturonate was something that we had chosen, when we said to the Army, okay, we want to take this challenge on, what are the different carbohydrates present in food waste? Galacturonic acid was very high on the list. It turns out it's found in fruit peels. And there was several other compounds that were high on the list as well. So our basic challenge that we had here was, could we actually engineer a system that would have stable, consistent output, even as the input was varying all over the place? Okay? So to do this, what we decided is that we needed sensors. And we needed sensors that would respond to the different things that might be in food waste, to only turn on a pathway to utilize one particular compound when its substrate was present, and leave everything off. And if both substrates were present, turn both of them on. So just very briefly, on the left-hand side is the sensor we already built for galacturonic acid. We used the exact same process to build one for gluconic acid, which was another of these compounds on the list. And what I'm showing in the chart here is just the fact that the galacturonic acid sensor responds only to gal and not to gluconic acid, and vice versa. The gluconic acid sensor responds only to gluconic acid and not to galacturonic acid. We actually then decided to build pathways from each of these compounds to glycerate. And we needed a different pathway, and I'll show you in a couple slides why we needed a different pathway. But we still are relying upon biology doing what it already does, which is that in this case, we're letting the cells utilize known and endogenous pathways to make a particular intermediate, and then we have to add 2 new proteins to get to glycerate. I'm going to skip through the next part fast, both because I'm running out of time and because it's only going to be comprehensible to a few of you, but because we talked about the need to knock out different genes. I'm showing you the results of that exercise, which is knocking out different versions of all these other ways that those molecules can be used. And you can see doing that gives us a lot of cases. We picked the best performer in that case. We also can do some other common tricks in metabolic engineering, which involves looking at how strongly the genes are being expressed by playing around with ribosome binding sites. That gives us some improved performance. And then we can also, because we have 2 genes now, play around with the order of those 2 genes, and that actually leads to the highest productivity that we could see. The reason we had a different pathway is that now I'm showing you adding gluconate to the system, and I only need 1 additional enzyme in order to make now the same compound from 2 different products. I'm not going to show you all the details of that. I'm just going to say that this pathway is now regulated based on gluconate presence using this gluconate biosensor. We did a similar sort of exercise to identify what's the best activity we can find from there. And in this case now, what we did was to start with a system where we had each pathway individually. That's the data that's shown here. We have almost 2.5 grams per liter from galacturonic acid, a little bit more than 1 gram per liter from gluconic acid. And then we put both of the pathways in the same cell, but then provided only one or the other of the 2 substrates. And something very interesting happened. We actually got less production from galacturonic acid, and we got more production from gluconic acid, but they actually started to even out. And we have no idea why. And then as we tried a different mixture of the 2 substrates, we actually found that they all pretty much evened out to the same amount. So we started with this objective of could we take multiple substrates and then engineer them to be consistent when the input was variable. And it turns out it did that on its own. We got essentially consistent output irrespective of the input would be. We've tried a number of ways to explain why this happens, and again, we really don't know. I am out of time, so I'm going to skip through the last example, and I'm just going to point out one thing we've been working on, which is to deal with plastics use. We had a talk about plastics earlier today from Jose Jimenez, and so I'm not going to repeat all of this. The bottom line is that we know plastics are a problem, and that accumulation of large amounts of plastics in the environment is pretty damaging. But what we also know is that we need plastics for a lot of the function that we have in our everyday lives. It makes our lives better. In some ways, it helps to preserve food and make it more available. In some ways, it helps to actually provide important medical devices. So what we've been working on with collaborators is how can we think about designing plastics that are built for their end use, or essentially built to be degraded. We've thought about this from the whole cycle. It starts with, can we actually identify monomers that we can produce biologically that can go into those plastics? I don't have time to go over this, but I will just say we have worked on this and have looked at being able to develop pathways to new kinds of monomers that will give us the functionality that we need in biologically accessible polymers without having to go through the traditional ways of using petroleum in order to produce them. And we actually rely upon nature for this as well. So we were inspired by a known pathway for degrading amino acids, but we decided rather than degrading those amino acids, we would be able to produce the precursors for them to make these compounds of interest. And so we were able to produce 2 new compounds. This work has not been published yet. It's currently under review. The titers are very low, and so now we have to switch into that mode I talked about before, which is how do we get more of something once we've shown that we can make it. And then the last part, this also sort of harkens back to a talk earlier today, is how to actually prove that these polymers are biodegradable. The well-known assays for doing this rely on single species, and they take a long time to do. So we're working with collaborators as well to develop high throughput ways to dramatically reduce the screening time so that we know we can get the materials that we need. We know they can be produced from resources that are more sustainable and environmentally friendly. And when we're done using them, we don't have to worry about them persisting in the environment indefinitely. I'm going to skip over the summary and just say biology is great. And acknowledge the folks who've done the work. Their names are highlighted here, and they were on the slides as we went throughout. I'm delighted to be here. I thank you for your attention and for your laughs at my jokes, and I'd be delighted to take any questions.
Thank you, Cristela, for a wonderful, absolutely wonderful talk. Yes, I'm happy to take a few questions.
It's late, but don't be shy.
Yes.
2 minutes.
Can I ask you to introduce yourselves as you ask the questions? It's easier for the speaker to know where the question is coming from.
Yeah, my name is Nick James. I'm an electrical engineer. I'm just curious, you mentioned plastics and you say they're needed in our lives. Can we move more, with, especially with food containers, to metal containers which are not oil-based, or maybe to glass? Milk bottles used to be in glass, not plastic. We were using, they work perfectly well, and they're recyclable much more easily.
Yeah, so it's a great question. So in this case, I'm talking less about, when I say food, less about containers for just transporting in the short term, but more about packaging for being from the point of harvest to preparation to the market. So what has happened over the years is that the shelf life, especially of produce, has increased dramatically as a result of specialized plastics and materials that have these barrier properties that prevent oxygen. So when I was a kid, you could not buy salads, lettuces that were already cut up, because as soon as you cut them up and put them into a package, the exposure to oxygen would cause them to rapidly degrade. So the fact that you can go into the supermarket and find rows of all different types of lettuces, and you can pick them up and they've been on the shelf for a couple of weeks, it's because of the specialized packaging that's there. So that's one of the examples of something that's had a pretty significant impact on the quality of human life. We still have tremendous amounts of food waste, but that innovation actually resulted in a dramatic decrease in food waste, because you could transport foods, fresh foods especially, in longer distances, and you could maintain them for longer when they arrive at their end use. And glass and metal won't do that. But you're absolutely right. It's easy to convert if you're looking at only having something for a day, take out containers, things like that. Absolutely, there are ways to do it. You still have to worry in some cases about reusability and making sure that you have reuse or that you have recyclability. Otherwise, we'll run out of metals at some point too, right? So all this ends up being about resource conservation. But the plastics for food are really about these specialized cases where we've been able to maintain the shelf life and make more foods available to more people because of the materials innovations.
Hi, Cristela. Thanks a lot for the talk. I loved it. I'm Jose, by the way. I don't think I need to introduce myself. Yeah, I just wanted to ask about the comment you made about low titers, and that it's better to produce something compared to not producing anything. So, that's the kind of argument that normally I tell to my colleagues that do scaling up, and they are typically completely unimpressed by the peaks that I saw them, the HPLC chromatograms. So how low is low, and what makes something really, I'm not going to say profitable, but something that makes sense to do for industry?
Yeah, so great question. So I would say the part I just raced through at the end, we had 2 different compounds, one that was about 50 milligrams per liter, one that was about 700 milligrams per liter. The closer you get to in our experience, the closer you get to 1 gram per liter, the more hope you have of being able to convert that into something that, quote, matters, right? But the reality is what you said you weren't going to say is what really matters, right? So how much you need to matter depends on the value of what it is that you're trying to produce. I talked about biofuels as part of the background because I have a history of working on biofuels. But the reality is that at the time I was working on biofuels, this is being recorded, okay? I should be careful what I say. All right, so let's say today. Let's not talk about the time I was working. Today, the cost associated with biofuels is just too high. Even if you are looking at environments where, in Europe, for example, the cost of petrol is much higher than it is in the US because Europe attempts to have a tax assessment that reflects what the actual cost is. And we don't do that so much in the US. But even though petrol here is several-fold more expensive than it is in the US, it still is cheaper than what it would cost in order to make a comparable biofuel from biomass. But it almost doesn't make sense to do that. The cost is one part of it, but when there are other alternatives, given the fact that biomass, by the way, is also going to be limited. Right? So there's lots of things that we're going to want to try to make from it. And the scale at which you have to replace fuels is just so high that by the time you do the scale and you do the cost, it makes sense only for those areas where you don't have any other options, but where you have other options, you explore them. Right? So in a case like that, it ultimately is going to come down to what are the real costs, because the costs are going to vary based on supply and demand, and where can you actually produce enough to make a difference. There is an entirely different set of molecules that have another wide range that go from specialty chemicals to commodity chemicals. And in general, that curve is inverted, where the higher the volume, the lower the cost. Okay? Those are not unrelated to each other, right? You can make a lot of something, or rather you can purchase a lot of something only if it has a low cost. And if it has a high cost, you can't purchase very much of it, so you don't make very much of it. So in part, it matters if you're looking at those very high value compounds, you can do the cost calculations and convince yourself that 20 grams per, actually, you can convince yourself incorrectly, in my opinion, that 5 grams per liter is enough, because I've heard people say that. But you can even get to 20, 25 grams per liter and actually have something that's sort of useful. But the further along that curve you go towards higher volume, then the higher your concentration has to be, because your separations cost, now I'm a chemical engineer again, right? Your separations costs are going to be inversely proportional to your concentration. So it's not that you can't make something at 20 grams per liter and sell it. It's that the cost to purify it compared to if you had 120 grams per liter is going to make your production costs so expensive that you can't do anything about it. So the markets matter is the overall message, right? In terms of how much you need in order to say, okay, I have something. There's a technical answer to that, and my technical answer is once you get to about 1 gram per liter, you have some flexibility to be able to push higher. But the ultimate answer in terms of what matters is all about the markets, whether or not what you're making has to be sold at very large volumes for very low cost, or whether you can do it for much smaller volumes at much higher cost.
Thank you. Yeah.
It's a big auditorium.
Thank you. Ellis O'Neill, University of Nottingham. It's a fascinating talk, and I think we're all probably quite sold on making stuff in biology here. But when you show an oil drum at the beginning and you're looking at just that little bit we turn into useful products,
yes.
How much waste are we going to be making with this engineering biology, synthetic biology, that we're going to have to process and deal with in some way?
Yep.
And yeah, we're taking CO2, turning it into glucose, turning the glucose into something, and maybe back into CO2.
Yep.
And purifying your compounds from water as opposed to from crude oil. Yep. You've got a thermal capacity of water. You've got a lot of stuff you're going to have to deal with at some point.
Yes. So great question. So this is the other one. When I said that people can convince themselves that 5 grams per liter is sufficient, but it's not. In those analyses, I think what's been forgotten is the cost to deal with waste. Right. So the first answer is you actually, if you're going to minimize the waste that you do have to get your titers, your concentrations up high enough that you can minimize the waste that you produced, right? So if you're minimizing the waste that you produce, then that's one issue. For biological systems, though, there's two parts of this that make them better than comparable waste when you're dealing with oil and gas. One is that one source of waste is actually the biomass. We have precedent for this, which is in the ethanol industry. Tremendous amounts of yeast biomass waste, quote unquote, that's being produced to make ethanol. It's not actually wasted. It's dried down and it's sold as animal feed because there's an extraordinarily high in protein content. Right? So we actually have a way of reusing the solid biomass waste. There's also ways you can hydrolyze it and sort of recycle it back to the system if you need in order to deal with that. So one aspect of the waste, there are other ways that you can use it because it's non-hazardous in a way that you can't use the waste that comes from traditional petrochemical refining. And then the other is that you're right, there's a lot of water, but because it is water, and it doesn't have organic solvents in it, your ability to be able to actually rehabilitate that water is useful as well. So there are studies that are being done to try to figure out the extent to which you can do some partial cleanup and actually recycle that water such that you're not constantly having to bring fresh water in. The last point, because you made a comment about the separations processes and potential. So biological systems, I'll put it this way, we can't distill everything. Right? So you're absolutely right. If the answer is it's going to be in water and we're going to use distillation the same way that we do with ethanol, it doesn't make any sense. The energy demand is going to be too high. The costs associated with that are going to be too high in terms of the CO2 footprint. But these kinds of systems give you additional possibilities. There are new membrane separation techniques that are being developed that are much less. We don't work on this. This is just things I'm aware of. There are new membrane separation techniques that are being used. And you also have the potential for new kinds of configurations. So I'll give you one example of a project that we worked on that we're back in the biology phase of it, but in which it had gone towards more of a process engineering phase. We were working with, Tom Ellis mentioned earlier today, non-model organisms. So we were working with a non-model microbe, something that had been isolated from a natural environment that is essentially an underground CO2 reservoir. And the carbon dioxide in that environment is under high pressure, so it's in supercritical form. Supercritical CO2 is actually considered to be a sustainable solvent. Okay? And so we had a collaborator who's an environmental microbiologist who sent a student to where this CO2 mine was, pulled up what are called formation fluids, so essentially the liquids that come out of that, and after several rounds of selection, isolated this microbe that will germinate and grow in the presence of supercritical CO2, which, by the way, kills most everything else. Right? So we came up with this great idea. A lot of great academic ideas, by the way, are magical, fantastical, right? Our fantastical idea was, okay, we're going to do two-phase fermentations. We're going to use water, which is what the cells need to grow in, and then we're going to have this supercritical CO2 layer. And the benefit of that was twofold. One was nothing's going to contaminate it because supercritical CO2 is going to kill everything else. But the other is that the products we were targeting to make would phase separate into the supercritical CO2 phase. It's immiscible in water. You could actually extract it pretty easily. And all you need then is a slight depressurization. And then whatever it is that we're producing water-soluble molecules, those crash out of solution. You repressurize the CO2 and recycle it. And we did modeling there to show that you could potentially reduce the separations cost compared to distillation. I think the number was something like 75%. So all of this absolutely takes continued innovation, but at the base level, because what you're working with is predominantly water and it's non-hazardous, we have a lot more options for how to deal with the waste.
Thank you.
Okay. Quick question.
Thanks very much for your talk. Gabriel Abrahams from the University of Oxford. Also in the panel before, Tom mentioned this idea that we should be using all our thousands of cells to get lots and lots of data. And so I guess I was wondering, in your talk, there was this general theme, I think, of kind of very lovingly and carefully handcrafting you look at what you want and what you have and picking the proteins to put into your cell that will create that pathway. And so I guess I was wondering if there's a future here where this kind of design can happen in some sort of high throughput way, or where you see that going in the future.
Yeah, so I'll attempt to answer it, and then you tell me if I didn't answer the question that you really asked. Okay. So I heard Tom say that, and I immediately thought of the place where we use that is when we were trying to do directed evolution. And I didn't talk about any of the work here, but we have some adaptive laboratory evolution experiments going on, actually in relationship to the project that I just talked about with supercritical CO2. And we've also had studies where we're trying to specifically evolve individual proteins. And in those cases, you absolutely are reliant upon all the diversity. You need millions of cells because you don't know which cell is going to be the progenitor for the phenotype that you're looking for that you can grow up in large amounts. So there are certainly certain use cases where we want to take advantage of that. And there are also cases where if we had a biofoundry, right, which I got to visit yesterday, then we would have the ability to scale down in a way that you could have the high throughput that's necessary for a lot of these different cases. When we are looking at generating diversity, that's when we focus on, can you do 1,000 experiments in one vessel? When we get to the point where our objective is to try to be consistent and robust, we want to do the opposite. We actually are trying to minimize heterogeneity as much as possible. There was a talk earlier today about heterogeneity as well. And we are, at times, we want to understand it to be able to really engineer around it. But our objectives, if our focus is ultimately on commercialization and translation, in that case, in the canonical case, I should say, you actually want to minimize the diversity. Now, there's a different case or different set of cases that have yet to really emerge where I think the exact opposite of what I said is going to be true. I gave this example of looking at food waste as a starting material to make compounds from. And everything I talked about today is work we do in E. coli, and I show this example of building these pathways in E. coli. I am under no illusions that E. coli as a host could be engineered to the point where you could have a single E. coli culture, and it's going to be able to take banana waste and hamburger waste and pasta waste and convert that into anything. But I do think you could have a consortium of organisms, right, that are each designed to interact with each other, where some of them are going to be activated and others aren't, based on what the input feedstocks are, and that you could engineer that consortium in a way that you would have consistent output. And one of the reasons I believe that is because it already happens. It's called anaerobic, it's actually called enhanced biological phosphorus recovery, EBPR, is a wastewater treatment process that has been in operation for decades that uses very complex communities, one set that's anaerobic, one set that's aerobic. It switches between them to make sure that you can consume all of the organic waste that you have there. And it works until it doesn't, right? But it'll work for a long time, and then it'll have an upset, and then you sort of restart it and it works again. So I think as we think about much more complicated inputs, when we get to the point where we're trying to address the waste remediation problem on the planet, and we think more about circularity, that the complexity of the inputs is going to require us to think about how do we have more complex processes, but it also means changing how we've, and I say this as a chemical engineer, changing how we've traditionally thought about those processes, how we characterize them, and how we design them for tolerable amounts of upset, right? Because the traditional way that you design a production facility is for it to run until you shut it down. But the more complexity we have in these biological systems, the more they are going to do whatever they do, right, without us knowing it, and we're gonna have to reset. So we're gonna have to design new processes. We're gonna have to learn how to better characterize them and learn how to operate them and with greater uncertainty than we're accustomed to doing now. Did that work, more or less?
Yeah.
Okay.
Amazing. Okay. I'm going to move questions to we have drinks afterwards, is that right? Yeah, so I'm going to, please, going to give Cristala a break. Please join me in thanking her for an amazing talk and discussion. Thank you. And before I invite you to join us for drinks, I also wanted to ask you to please thank amazing AV team and tech that allow us to record this. Everything has worked very well. Please thank the BSL interpreters as well, and I want to extend very special thanks to the IMSE team. They're somewhere around. Elena, have they just run out every year? Elena, Anne-Marie, Steph, and very special thanks to Leah, who is our events organizer. She's absolutely amazing, and she made this possible. She will be outside, so if you see her in the blue suit, thank her for this amazing day. And again, thank you, Cristala. Thank you all for being here. Join us for drinks.